Dear ladies and gentlemen, esteemed colleagues, it's my great pleasure to welcome you to this first session and symposium on the immune system in the context of sepsis, which we consider as a double-edged sword. And it's my great pleasure also to thank all those who have contributed that we came that far where we are. And part of these supporters are also the Berlin Medical Society, and from the very beginning, also under the leadership of Eva Roth, but also after Claudia Spies from the Charité has taken over, were supporters of the Global Sepsis Alliance and also the Sepsis Foundation. And I can tell you that we have made huge progress, especially over the last year, especially in terms of stronger involvement of WHO and also policymakers from around the globe. Along these lines, before I announce the welcome words or the greetings of Dr. Tedros, I would also thank our sponsors, especially the German Ministry of Health, who via this Germany Recognizes Sepsis campaign, which comprises 5 organizations, supports annually this kind of events with 25,000 euros, and I want to thank this and make this aware. And now I would like that we present the video greetings of Dr. Tedros, which yesterday have been also presented at a meeting in the Bundestags with over 50 participants, including quite a number of MTBs, but also other supporters from industry and from society.
Dear colleagues and friends, Guten Tag allerseits. WHO congratulates the Global Sepsis Alliance on the launch of the 2030 Global Agenda for Sepsis. This is a valuable tool in our shared work to prevent the suffering caused by sepsis, which kills 11 million people every year, including children, older people, and other vulnerable groups in low-income countries. The global agenda is well aligned with WHO's work on sepsis. We have made good progress in raising awareness about sepsis, but we can and must do more. Millions of lives can be saved with proven, affordable interventions that are feasible in every context. WHO is supporting countries to improve the prevention, early recognition, and timely treatment of sepsis. We're implementing the Global Action Plan on Infection Prevention and Control. We're developing new guidelines on clinical management, and our sepsis toolkit supports communities and countries to strengthen emergency and critical care, improve outcomes, and save lives. We thank the Global Sepsis Alliance for your close partnership and leadership on this issue, and we're deeply grateful to Germany for its long-standing support. Vielen Dank. We look forward to our continued partnership to reduce the preventable burden of suffering caused by sepsis.
So we should applaud Dr. Tedros, I think so. And before we move to the next greeting, I really wanted to thank also our co-organizer, Professor Stefan Kaufmann. And it's another sign that we have made huge progress over the last years to be co-sponsored and to do this jointly, not only with the Berlin Medical Association and the Virchow Foundation, but also with the most prestigious Max Planck Institute for Infection Biology, which has been led for a long time by Stefan Kaufmann. And we are so grateful that he also took over the task of our ambassador on vaccination. Thank you again, and please let us hear the words of Hans Kluge, who is the WHO Director for Europe.
Sehr geehrte Kolleginnen und Kollegen, vielen Dank für die Gelegenheit, heute zu Ihnen sprechen zu können. Sie haben von der Bedrohung gehört, die die Sepsis weltweit für die Gesundheit darstellt. Werfen wir nun einen kurzen Blick auf die Situation in Europa. In der europäischen Region der WHO sind Jahr für Jahr etwa 3 Millionen Menschen betroffen. Fast 7000 Menschen verlieren unnötigerweise ihr Leben. Von Sepsis sind Frauen, Kinder, Senioren und Menschen mit chronischen Erkrankungen unverhältnismäßig stark betroffen. Sie ist eine ernste Bedrohung für die öffentliche Gesundheit. Doch, und das ist ehrlich gesagt erschreckend, in vielen europäischen Ländern steht die Sepsis-Problematik immer noch nicht weit genug oben auf der Tagesordnung. So haben nur wenige Länder die 2017 von der Weltgesundheitsversammlung verabschiedete Resolution zum Thema Sepsis vollständig umgesetzt. Das muss sich dringend ändern. Unser Aktionsplan ist die Globale Sepsis Agenda 2030. WHO Europa befürwortet uneingeschränkt ihre 5 Schlüsselbereiche: Handlungsbereitschaft der Gesundheitssysteme, Einbeziehung der Bevölkerung, Forschung und Innovation, bessere Vorbereitung auf Pandemien und Notlagen sowie als grundlegende Voraussetzung Führung durch die Politik. Vor kurzem hat WHO Europa in Griechenland ein Fachzentrum eingerichtet, das auf eine Verbesserung der Versorgungsqualität und Patientensicherheit in der gesamten europäischen Region hinarbeiten soll. Dies ist ein entscheidender Schritt nach vorn, damit unsere Gesundheitssysteme in der Lage sind, Sepsis wirksam zu verhindern, zu erkennen und zu behandeln. Und es wird auch anderen Ländern in der europäischen Region als Vorbild zur Nachahmung dienen. Sehr geehrte Kolleginnen und Kollegen, die Covid-Pandemie hat uns schmerzhaft vor Augen geführt, wie unvorbereitet unsere Gesundheitssysteme sein können. Die Globale Sepsis Agenda 2030 wird dazu beitragen, dass wir besser auf den Schutz besonders gefährdeter Menschen vorbereitet sind. Es liegt auf der Hand. Zur Verringerung der Zahl der Sepsisfälle ist eine gemeinsame Anstrengung erforderlich. Deshalb appelliere ich dringend an die Regierungen und ihre Gesundheitsministerien, an die Gesundheitsberufe, an Wissenschaft und Gesellschaft, hier an einem Strang zu ziehen. Vor allem müssen wir dafür sorgen, dass Sepsis-Überlebende und die Familien, die Angehörige verloren haben, am selben Tisch sitzen. Ihre Erfahrungen müssen uns den Weg weisen. Zusammen mit meinem Team bei WHO Europa stehe ich bereit, Sie in Ihren Bemühungen zu unterstützen. Ich danke Ihnen.
Before I hand over to Stefan Kaufmann to moderate the session, I would like to welcome also Dr. Mariam Yashi, who, as a former Minister of Health in Georgia, knows what is necessary to get major health threats on the political agenda. And you just heard that is not yet the case, despite the adoption of the WHO sepsis resolution, where Germany, jointly with other German-speaking and other countries, played a crucial role. And Mariam really has made the difference by kind of knowledge, this was the missing gap, and she closed it. Mariam, please move forward, and she will address 2 of these 5 key steps that we need to address and to move forward, because they helped to be successful in fighting other global health threats. Mariam, please.
Professor Kaufmann, Professor Reinhardt, esteemed colleagues, it's an honor to share with you some of the highlights from the 2030 Global Agenda for Sepsis that we launched yesterday at the German Parliament in partnership with the Global Health Subcommittee of the German Bundestag, Unite Parliamentarians Network, Sepsis Stiftung, and the Virchow Foundation. This document is the first multi-year global strategy for sepsis that was developed with engagement over 70 multiple stakeholders. The strategic planning process started with sepsis side events organized for the first time in parallel to the United Nations General Assembly last year, as well as the 2023 World Health Summit in Berlin, and most recently in Geneva on the margins of the 77th session of the World Health Assembly. And we were proud, together with Sepsis Stiftung, to partner with Unite, with the Virchow Foundation, Clinton Health Access Initiative, Medical Women's International Association, and the Global Antibiotic Research and Development Partnerships for the strategic multi-stakeholder dialogue sessions. In addition, the development of the document that will be publicly available through the Global Sepsis Alliance website, as well as the Sepsis Stiftung and the World Sepsis Day websites. As of today, we received contributions and endorsement from 29 multiple stakeholders, both public, private, civil society, and organizations representing sepsis survivors and their families across the 6 regional sepsis alliances, starting from Africa, Asia Pacific, Europe, Latin America and Caribbean, Eastern Mediterranean, and Latin American Sepsis Institute. More importantly, we received official acknowledgement from the World Health Organization that the staff of WHO's Geneva headquarters and the regional offices contributed to the development of the global agenda for sepsis. The document consolidates the latest evidence on the global burden of sepsis, starting from human, societal, health sector, and economic burden, achievements over the last 2 decades of the global sepsis fight, including the evolution of the clinical knowledge, global advocacy, and success stories from 15 countries, mostly from high-income states that have already developed the national action plans and evidence-based protocols for the sepsis management. The document summarizes policy, institutional, community, research and development, and measurement and accountability gaps, and provides the 5 strategic pillars as the way forward. Of course, the document addresses the immense human burden of sepsis affecting almost 50 million children, women, and men worldwide, and taking over 13 million lives based on the latest estimates from the Institute of Health Metrics and Evaluation. The facts are known. The 85% of the global sepsis burden are affecting the middle and low income countries, 40% of cases in pediatric populations, and sepsis remaining still 1 in every 5 deaths, the primary reason. The document affirms critical importance of sepsis vis-à-vis the 2030 UN Sustainable Development Goals. And while the document explores comprehensively the health related SDGs and 8 additional Sustainable Development Goals that sepsis is closely linked, we would like to highlight that just 2 targets related to the theme of today's symposium. Target 3.8 on universal health coverage through accessibility and affordability of essential medicines and vaccines is directly related to the sepsis prevention agenda, and target 3.b on research and development of novel vaccines, diagnostics, antimicrobial treatment, and immunotherapies for sepsis would be also the critical link to accelerating progress towards the 2030 agenda of the United Nations. The 5 strategic pillars of the new global agenda include political leadership and multilateralism. Without meaningful and consistent political leadership, we would not be able to make any difference in the global fight against sepsis, and that is why the document will call for the establishment of the high-level political platform with engagement of policymakers, legislators, parliamentarians, government representatives, and heads of states. If we really are ambitious to make sepsis the next success story of global health, we definitely need historic breakthrough in political and financial investments. Health system readiness for sepsis and its sequelae is the second strategic pillar, followed by whole of society approach to improve sepsis literacy and awareness as a medical emergency requiring immediate care-seeking behavior. Sepsis research and innovations, and sepsis in pandemics and other emergencies are the 4th and 5th pillar of the document. In view of the time limitations, of course, we will not be able to go through the detailed strategic pillars, but we would like to highlight some of the priority directions across the 5 strategic pillars that relates to the double-edged importance and role of the immune system in fighting sepsis. Under the strategic pillar 1, countries, 80% of high-income countries and 50% of low and middle-income countries are called to establish national action plans for sepsis, either standalone documents or as part of holistic national health sector plans and policies. They are also called for integrating sepsis response into the basic national universal health coverage packages, including infection prevention and control and immunization schedules. Immunization as the sepsis prevention measure is highlighted across the strategic pillar 2 and 3 of the document, emphasizing the importance of the 2030 immunization agenda for attaining at least 90% of a basic routine immunization coverage against children, from the current baselines of 84% for the diphtheria, tetanus, and pertussis pertaining vaccine, and rotavirus coverage of only 51%. The document also calls for effective coverage of immunization among high-risk groups, yet the baselines, for example, from Spain, indicate that over 65-year population groups are immunized against influenza in 54% of cases, and only 26% of healthcare workers are protected from the influenza virus. Over 50% of the community representatives are expected to understand the importance of immunization in preventing sepsis as one of the strategic measures. Yet, data from Canadian scholars indicate that only 25% of general public understand the importance of vaccination in preventing sepsis. Finally, with 120 armed conflicts and more than 662 million children, women and men living in humanitarian crisis, reaching the affected populations with vaccination would be critical for protecting them from the increased risks of infections and relatively sepsis. Finally, the document calls for establishment of the Global Sepsis Innovation Platform that would be the public-private partnership of scholars, innovators, patients and families affected by sepsis, based on the experience of the US Sepsis Alliance, and we hope that GSA, US Sepsis Alliance and Sepsis Stiftung will join the forces for establishing the platform. And the innovations platform will discuss consistently the unmet needs in sepsis prevention in terms of novel vaccines against the pathogens, leading pathogens in the sepsis etiology, diagnostics, including biomarkers, molecular point-of-care solutions, as well as artificial intelligence tools, and therapies, including antimicrobial, immunomodulatory, precision medicine, and theragnostic tools. And the document also calls for major investments, both from public and private sectors in the sepsis science, building on the excellent examples of US BARDA, that in 2024 expanded its collaboration and support to host-directed therapeutics research for immunomodulatory innovations for improved clinical outcomes of sepsis. So as noted, you will be able to access the full document on the website, and we look forward to closer collaboration. Thank you.
Without further ado, as announced, I would like to ask Stefan Kaufmann to deliver his presentation. And please take over and then to move forward, because we are already somewhat short in time, but it's worthwhile, and we have also room for discussion, and which is important.
Morning, and thank you so much. Can you just have the first slide, or do I do anything? Yes. Well, I want to talk a little bit, or give a little bit of an overview on vaccine for sepsis prevention. And obviously, I come from the vaccinology field on infectious diseases, but I realized that sepsis is even more complex, and we'll come to that. If you look at the success of vaccines for a high-income country like the United States, over the last 30 years, more than 1 million deaths had been prevented, and the economic benefits, and I would like to stress that, also I'm a basic researcher, are enormous. More than 500 billion in direct cost had been saved. If you look at the EPI, the Expanded Program for Immunization, as an example for low middle income countries, you realize that over the last 50 years, more than 150 million deaths had been prevented. So globally, and roughly speaking, you can say that about 3 million deaths prevented every year, and for $1 spent into vaccine research and development, more than $40 will be returned from the investment in direct and indirect cost. This is all roughly, but it gives you a kind of nail-of-thumb idea about it. And as was just appeared last days, actually, in the Lancet journal Respiratory Medicine, another 1.6 million lives had been saved through vaccine over the last 2.5 years, caused by COVID or prevented in the case of COVID. And obviously, RNA vaccines were instrumental in that, and we will hear more about that. And that contrasts that. We heard it already. Just let me say it again. Some 48, 49 million cases annually are caused by sepsis, and more than 10 million and more than 10 million deaths annually. So vaccination has contributed a little bit to reduce this kind of enormous burden, but there could be done more, and that's my true belief. Now I will give you a very short overview separating vaccines in whole cell vaccines and subunit vaccines. We'll not go into any details of the current vaccines as they are indicated here, but rather give you a kind of my ideas, which are biased by myself, but on the principles, what vaccines could be developed in the near future and how that should look like. So my agenda for today is we'll talk a little bit about sepsis, then a little bit about principal mechanisms of vaccine-induced protection or immunity, major vaccine types, and then have an outlook. Let's start with the sepsis thing. We all are aware there is an issue with sepsis because it's not a single pathogen disease. Multiple pathogens can be the agents that then later cause sepsis, though there's a plethora of pathogens. And I would argue, and we may discuss that later, that for some pathogens there may be specific vaccines that are better suited than for other pathogens. So I would argue we need different vaccine types because also that is a very complex situation. Some situations are more simple, some are much more complicated. And as some research and development against sepsis is complex, I realize that, but I would argue that vaccines to prevent sepsis should be possible in many cases, at least, if not all, provided there is the will to do that. As you know, there are about 70 to 80% of sepsis cases are caused by bacteria, and I will therefore focus on bacterial-caused sepsis, also because I know more about bacterial infections than viral and other infections. Number one clearly remains Staphylococcus aureus, followed by Escherichia coli and Streptococcus pneumonia. And I would just like to mention here that in red you see the two pathogens which we have vaccines for already, prepared and developed for these diseases, but also as a consequence helpful for septicemia, for septic cases. And what you see also here in dark red is they are real bad bugs. They are troublemakers because almost all of them or all of them are multi-resistant. Not all of them, of course, but a large proportion of these pathogens are multi-resistant, and therefore this is a particular issue. And just to remind you, vaccines have a hidden bonus. Vaccines can also reduce the development of antibacterial resistance, has been shown most nicely in the case of sepsis cases by pneumococcal vaccination, where both in high-income countries and low-middle-income countries, there was a significant reduction in antibiotic resistance development or emergence caused by the introduction of vaccines. So let's a little bit look at the vaccine types, and I really will focus today on active vaccination, preventive, pre-exposure, but in sepsis we also have to consider post-exposure because a number of microbes will persist in the pathogen and cause sepsis later, and I will focus on the classical kind of vaccine types, that is a vaccine directed at the pathogen. Later in the discussion we will come to HDT, host-directed therapy, which is covered by others. In my opinion, I will give you three different types of vaccines. Vaccines against a single pathogen which has a single protective antigen, a protective antigen defined as an antigen that causes antibodies that are neutralizing and therefore protect. I will then go to the next step, vaccines against a pathogen like tuberculosis, where you have a chronic infection, a single pathogen, but multiple antigens, and where there is no idea whether a protective protective antigen or group of antigens exist, and this is, in contrast to the first type, is based on T cells, the protection, whereas the acute ones are based primarily on antibodies. And then we will come to sepsis and we'll see that both situations may arise. So different vaccines really need a different mold, and a different pathogen also needs a specific type of vaccines. This is a simple scheme of what we will perhaps hear much more. The case of an acute infection caused by a virus which has a single protective antigen, that is that B cells which produce antibodies cause neutralizing antibodies. There are other protective mechanisms, but this is the central mechanism, protection through neutralizing antibodies. And here, as we will hear more, RNA vaccines are now really becoming the front runner if they are not already the front runner. There are only few antigens in this virus, 14 open reading frames, and a single one is sufficient. So this how it looks like. In red you see the unvaccinated situation. You develop disease, you die, or you survive and come back later. Most of these vaccines can prevent infection by neutralizing, in that case, the attachment on the host cell. And that is actually not clear how often it happens. In yellow, you see what I believe is more frequently happening, and that is the vaccine prevents stable infection, also then by the help of antibodies induced by the infection per se. And then you can either, and that's fine because then you are healthy, no virus, no agent is there. And then, of course, vaccines can also ameliorate disease or even prevent disease, but still with a certain microbial load and pathogen load in the host. And that has consequences for the disease development, prevention of disease or attenuation of disease, amelioration, and also reduced transmission, POT. In the next case, it's a little bit more complex, and I will not go into details, just illustrate here. Antibodies have no major effect that is known, and I would say there is no major effect. I don't exclude them, but it's the T cells that do the job, and T cells are living organisms, if you want to say it that way, and therefore T cells do not do that in the same way as antibodies. There is a fine-tuned biological mechanism that will induce a certain degree of protection, which often just means containment of the pathogen, and we will see, I would argue, Staphylococcus aureus, as we now know, is a little bit of like that type. Also, this is the most extreme one. So we have a long-lasting latency by the T cells, chronic disease if it develops, and it's about 4,000 protein-encoded genes, and we really do not know whether 1, 2, 3, 4, 10 are sufficient. It's very, very difficult, and that is the issue we have here. There's no neutralizing antibody. There is a little bit of an effect. T cells, therefore, are essentials, and as a consequence, there's nothing like a surrogate or correlate of protection like neutralizing antibodies, which you can test in vitro assays like it's done every year for influenza. So this is how it looks like here. You have this long-lasting, in gray, latent infection, and actually people in the field of tuberculosis like myself would be happy if you could keep latency. Would be better to eradicate it, like in yellow here, but you would be already happy if you could prevent disease, but leave the pathogen kind of be in a dormant stage. So it's the same thing, but it's a long-lasting thing. Here we talk about years. And finally, sepsis, I would argue you have both cases. There are pathogens like the pneumococci, and therefore we have a vaccine already there, acute infection, a single protective antigen with a variety of different carbohydrate structures. But in principle, if you have the right one, then it works. But there are also other ones, and in gray here, I would not say only infection persists, but it could also be colonization that persists. That is the pathogen or the kind of a microbe in the host, as long as it's harmless, can persist without a major immune response, and then later disease occurs. So I re-emphasize here, I think, in sepsis, you have both situations. Some are easily or more easily tractable, and others are much more complicated. Let me now go very briefly to the different vaccine types, and I skip a few slides. Somehow I was in the impression we talk about 30 minutes, but it's fine, I'll be going. So we have first, I say it again, the subunit vaccines. They are good for acute viral infections, some chronic viral infections. They are good for toxin producers, diphtheria, and they're good for encapsulated bacteria. And one, of course, would be one type would be protein adjuvants. And here we have an enormous progress in the case not of the antigens, but rather in the vaccine adjuvant development, very important step. We have the carbohydrate conjugates. This was one of the major discoveries in vaccinology over the last decades, before the RNA then were the real breakthrough, that you can attach carbohydrates to proteins and then get an immunogen that induces immunity, viral vectors. I would like to point out that in some areas on this globe, outer membrane vesicles are much more favored than we have it in Europe, in my opinion. You may argue against that. And of course, then we have the kind of breakthrough with the mRNA. The risks are also obvious. There is the possibility of evasion. If you are directed at a few epitopes, of course, evasion, that is either replacement or mutation, and then selection can occur. And there is, of course, a number of pathogens where there is no protective antigen, and therefore no single antibody or not a single group of antibodies then can do the major job. Let me skip that actually. So we have the antigen adjuvants, we have the conjugate vaccines, which are important for vaccines against sepsis agents. But I would like just to stress here that the adjuvant field, which is so essential for protein antigens and protein-based vaccines, has made enormous progress. We started with aluminum hydroxide in the 1920s, more or less 100 years ago, but now we have liposomal formulations or emulsions, and those really then benefit from the addition of pathogen molecules which are recognized by our pattern recognition receptors. Remind you of the Nobel Prize on that topic a couple of years ago, be it nucleic acids of different types, or be it bacterial cell wall components. Some of those components which may be dangerous in certain situations like LPS, but like the monophosphoryl A can also be good stimulators which improve the adjuvants. And I, without bias, because I'm not involved any in that work with JSK, the adjuvant system of JSK was a real breakthrough in the adjuvant field for a number of different vaccines, notably the papillomavirus and herpes zoster vaccines. Okay, then I would just mention here, for the kind of taking it to the records, I think that outer membrane vesicles, as they have been used successfully with type B meningococcal meningitis, is something we should have more intensive look into, because they have some promise whenever you need a couple of antigens which are in the outer membrane of a bacteria, and because you can also make recombinant vaccines, you can also apply that not only for gram-negatives, but also for gram-positives. So in principle, without going into details, a vesicle is formed from a budding of the outer membrane, and these vesicles can be isolated nicely and can be used for vaccination. And I will not go in any details. Of course, we have the RNA vaccines. We will hear more about those their success story. And my argument, I'm happy to discuss that then, is how many antigens and how the selection of the right antigens for more complex organisms will be achieved and can be achieved. But I would say when you use a subunit vaccine, there is clearly mRNA vaccines are on the top list of choices. And obviously also in the mRNA vaccines, it's not only the antigen that is important. You need a carrier that protects the RNA and also has some adjuvant effect. And protection needs to be in the bloodstream, but also in the cell, because once you enter an endosome, that's more or less the end of the RNA vaccine. So these wonderful kind of lipid nanoparticles that enclose the RNAs are, I would say, doesn't matter, but it's, I mean, equally important to have the right choice. And it was actually, in my opinion, really serendipity also to have both strategies together at the same time for the COVID vaccines. Now let's come to the second part, whole cell vaccines. Here, as I mentioned already, in the case of TB, we know that pathogenicity depends on multiple factors and molecules. That's true for some chronic viral infections, and some bacterial infections you really, at this stage, you really would also not exclude. I would say you may even bet on whole cell vaccines. They come as inactivated ones. I would be arguing that those are only, you would only go forward with those if you have a good adjuvant to combine them. And then there are different forms of viable vaccines that contain all antigens you need, but obviously they will also contain some products, gene products that might cause adverse events, be it autoimmunity, also that was less frequent than sometimes feared, but also obviously you have their pro and con again in both cases. I'll skip that again, and I would like just to mention here, because that's also my own expertise, you can obviously also improve viable vaccines. There are clearly cases now that has been approved to get stronger immunity by those vaccines. So let me come to the very last two slides, and that's the kind of outlook. I would argue for Staphylococcus aureus, one of the top agents for sepsis cases, there is hope, even though that 2 vaccine phase 3 trials had failed in the recent past. If you think of subunit combinations, perhaps even heterologous prime boost with viable and subunit vaccines, but I would think that Staphylococcus has a number of similarities to this chronic bacterial infection type. It's worth to go into it, and I think success could be possible. Strep pneumonia has been more or less solved. We know, of course, that you need new replacement strains come up. You need new combinations, but that can be done. Strep pyogenes, I would also argue, is possible. Most focus is still on the M protein. There are some hurdles, regulatory hurdles on that, but that could be solved by going into epitopes and peptides or RNA fragments, encoded fragments. Strep agalactiae and Enterococcus. Enterococcus is one of those where I think, even though it's gram-positive, outer membrane vesicles may be worth to look into it. Then on the negative side, clearly the Neisseria and Haemophilus vaccines exist, and here we could get improvements again, as said before, to be state-of-the-art. And Klebsiella pneumonia, again, I would think that a vaccine could be made probably again by combining a kind of subunit vaccine conjugate plus outer membrane vesicles, for example, similar also by the case of Pseudomonas. So in science, there's a lot of discussion about incremental research and disruptive research, and research development in vaccinology often has been considered very, very incremental, but we have seen with the RNA vaccines that disruption occurs and that therefore something can happen very rapidly. I'm not saying that this happens every 5 years or 2 years or whatever, but the chances are. I normally separate that into 4 forms, namely known known, known unknown, unknown known, and unknown unknown. I think a known known is that there is a solution by incremental research for obviously for S. pneumoniae, where you need replacement vaccines. That's a known strategy. If you can do it for this carbohydrate, you can also do it for that carbohydrate. Known unknown, I argued already that for S. aureus, P. aeruginosa, there is an incremental research that is based on vaccines that are already there, that that can come to a solution. More difficult would be the unknown known. Here I would think, and that is now beyond the vaccine field, that by immunologic modulations, e.g. vaccines, host-directed therapy, microbiome can be modulated to go on. And finally, the unknown, then really the difficult ones are when to get real-time personalized immune modulation. This is the kind of where a disruption has to take place. I'm optimistic, but this is the final step, and I thank you for your attention.
So much, and I think for at least the community I can speak for, this was really filling a knowledge gap in more depths, and this will help us to campaign jointly with the whole spectrum which is needed to decrease mortality. So it's complex, but knowing scientists like you and your community is a major breakthrough. Just one minor comment. You mentioned about 10 to 15% viral sepsis. There's data from our colleagues from Boston who assume, because we are underdiagnosing viral sepsis, they argue it might be at least in the range of 30%. So, but without further ado, I would like and thank you that at very short notice, and again, due to the fact that an esteemed doctorand of Stefan has been working with that, we were able at short notice to gain Dr. Ruben Rissi to address their experience and their learnings, and this was disruptive, and without disruption, mankind would never have made any progress. So,
thank you. Thank you so much, and thank you for inviting me. It's a privilege, obviously, and an honor being here today. While the slides come up, I'm just gonna 2 seconds to introduce myself. I'm Ruben Rissi, used to be a physician by training back in the old days. Always worked in regulatory affairs in the industry, and I joined BioNTech in 2019 in the regulatory affairs team, and for a couple of years, since day 0 to the end of 22, more or less, I've been the global regulatory lead for our COVID-19 vaccine development. So what I will try to do today is share a bit of that experience in developing a pandemic vaccine over the last few years, and maybe together we can understand what learnings we can take from that and how that experience can help us to improve, first of all, our ability to respond to pandemics, and second, other global health settings, and sepsis is one of them. And there are a lot of things that the pandemic framework for developing vaccines and therapeutics in a pandemic setting have in common with the world of sepsis and also other areas in infectious diseases, actually. So obviously, I speak for myself, and I am biased because I'm part of BioNTech, that's clear. So let's start from our COVID-19 vaccine development, and I'm gonna try and go very quickly to some of the part. You know, I work in regulatory affairs, so there's going to be some regulatory content there. I promise I will try not to be boring. I will be quick. You find references in a lot of slides, so if there's something that you want to look into, you want to read something, you find almost always a reference at the bottom to a publication, a website, whatever, so you can deep dive into that even if I go through the slides very quickly. So the development of COVID-19 vaccine, we called it light speed. Why light speed? This is the overall timeline of the initial COVID-19 vaccine, so the original vaccine, and you start from the identification of the genetic sequence for the SARS-CoV-2 virus, and at the end you have the initial approvals and the global rollout of the vaccine. I don't want you to look into all the different milestones here, but I would like to focus on 2 milestones. The first one is the start of clinical trials for the first vaccine candidates, and the last one is the approval. So the first clinical trials started here in Germany in April 2020, and less than 8 months later, we started having the first regulatory approvals for the vaccine. The timeline is even shorter if you look at what happened with the variant-adapted vaccines that started becoming a thing in 2022. So what you have here in the blue arrow is actually the 2 months that went from the recommendation that the regulatory authorities issued to switch vaccines to a bivalent BA.4/5 encoding sequence, and the actual time for approval of these vaccines. This was 2 months And I'm going to tell you at the end of the presentation how that was possible and how this is actually working still today. Because in 2023, the timeline was pretty much the same for the adapted XBB.1.5 vaccine. So 2 months from the time of the recommendation to switch the composition of the vaccine to the actual approval of the vaccine. And again, this year for the JN.1 vaccine that has just been approved recently. I could make a comment that there's another recommendation in the US for this year for a KP2 vaccine. This is probably a topic I will not go through today, which is about regulatory harmonization and alignment for recommendations in pandemic and post-pandemic settings. But I think that's a bit too much. So let's just focus on the timelines. 2 months for an adapted vaccine and 8 months for a completely new vaccine based on a new platform, it's very short time. And anyone with experience in drug development or vaccine development is going to be able to say this is very short, very accelerated. And obviously we got many times the question, how is that possible? There was a magic spell, there was a secret, how could you make it? And I always think about the example of the pyramids, right? Because we see something which is huge, incredible, and we keep asking ourselves, how could they build the pyramids with the technology they had thousands of years ago, with the tools they had? And the most obvious answer is always aliens, right? So this is how they built pyramids. Aliens helped them, or aliens built the pyramids in the first place. I don't know how many people believe that, but too many, probably. In reality, it's a very similar situation. The real answer is significantly simpler. It's a lot of hard work. And it's not a lot of hard work in the case of vaccines for us only, meaning BioNTech, Pfizer, Moderna, J&J, all the companies and the developers. But it's a lot of hard work from a number of stakeholders. It's the industry, that's true, but it's also academia, it's also researchers, it's clinical sites, it's governments, it's regulatory agencies, manufacturing facilities, testing sites and labs, hospitals. So there was a very strong commitment of resources in any possible definition from a large number of stakeholders globally with the same objective. And this is what makes these timelines possible. You need this commitment and you need this type of collaboration at global level. And this is something I will repeat again towards the end. Now, in the next few slides, there's a list of, I would say, a description of the regulatory framework that was pre-existing COVID-19 and that was used to facilitate and accelerate to a good extent the approval of COVID-19 vaccines and also therapeutics. I'm not going to the details, so I'm just going to quick through the slides. This is about the existing regulatory tools and pathways that were already in place based on previous epidemics and pandemics. And this is a mention of important global stakeholders that helped aligning and harmonizing the regulatory framework globally or taking responsibility for some pieces of the development of vaccines. I want to mention only one thing from this slide that is not very explicit, but we started our clinical trials in Germany in 2020, being a German company that was kind of obvious. The Paul-Ehrlich Institute was the regulatory agency responsible for the review and approval of the initial clinical trials for our vaccine. And they did an incredible job in providing guidance and advice in the early phases of development. So this is one of the enablers to get the clinical trials to start already in April 2020 was really the work of the Paul-Ehrlich Institute. So for the next pandemic, right, this is what we want to discuss. How can we be better for the next pandemic? So my first comment is let's hope it doesn't come. We know it will. It's a matter of time because this is how things go. But we always need to bear in mind it comes at an incredible cost. And when we celebrate the successes of science and there's new technologies and we have made something incredible with developing vaccines and also therapeutics in such a short timeline and so on, we need to bear in mind there's a huge burden in terms of human lives and disease that was due to the pandemic. So it's always a tragedy. Let's hope we don't need to go through that again, but we need to be prepared. What did we learn from COVID-19? First of all, we can develop vaccines quickly. This is something that it was not so obvious before, but we can really, nowadays we can really develop vaccines quickly. Doesn't mean it's going to be the same for every single pathogen, but technically this is something we can do in a very short timeline. We also learned that there's a number of issues in the regulatory space that are delaying or that are preventing the rapid development and approval of vaccines. There are still inconsistencies in the different regulatory requirements, inconsistent formats, administrative burden is still a thing, even within the European Union, let me say. There are still divergences in the different member states and their approaches and their recommendation and what they request and not. So this is something that we certainly need to address, at least from a pandemic preparedness point of view. Now, let's take a step back and try to think, from this experience of COVID-19 vaccines development, what is the easiest way to take this experience into something tangible for other areas outside of COVID-19, right? The most obvious thing you can imagine is that you can use the same platform of messenger RNA, the same format, and use it to develop vaccines for other infectious diseases. This is obviously something that we are doing. What you see on the screen is just a very simplified version of a messenger RNA when you have your coding sequence for a spike protein, for SARS-CoV-2 in the first line, or an antigen for Mycobacterium tuberculosis, or HSV, HIV, whatever. So this is obviously something that can be done, and it's something that we are doing. And it's obvious because we have achieved the proof of concept with the messenger RNA technology for COVID-19, and we feel there's a responsibility to use this tool that we have to try and explore as many therapeutic areas as possible to see where this tool can really make a difference. It shouldn't be a surprise that we're looking into some, I don't want to call them neglected diseases, but I would say unmet global health needs. So you see on the screen, we're working on malaria, we're working on TB, we're working on Mpox. Again, these are global unmet needs. They're not obvious diseases to address for a new vaccine, especially for a new vaccine platform, but they represent a huge unmet need. And again, I go back to the topic of feeling a sense of responsibility to make the best use possible of the tools that we have. And these are diseases that have a huge global burden, so we're trying to explore what mRNA can do in these areas. This is just a more formal overview of our infectious diseases pipeline, but I can skip it. Another topic for messenger RNA is not just the ability to adapt rapidly to new antigens that can be encoded without changing the platform, but it's also its ability to be manufactured in a relatively slim facility. So differently from some traditional vaccine manufacturing sites, you don't need the same footprint, you don't need the same complexity. So this means that you can create relatively simple and small manufacturing unit. In this case, we call them BioNTainer. It's a modular manufacturing unit that can be deployed, I don't want to say easily, but relatively easily without having a huge framework around it. And this goes also in the direction of ensuring that we can have manufacturing capacity and flexibility for potential epidemics and pandemics. But these BioNTainers can also be deployed in areas and regions where normally vaccine manufacturing would not happen. And this is what we're doing with Rwanda. We have a BioNTainer in Rwanda that has been set up, and we are working with the local authorities to make sure that that BioNTainer can become fully functional. This is a way, first of all, to make a new technology available in areas where it would not be available in terms of building hands-on experience, fostering research, but also, for example, helping local authorities to get an understanding of the new technology and to build the expertise in their home. And on the second hand, it may give you in the future, it can be a first step to really create a decentralized manufacturing capacity that can really help when you have local or regional outbreaks, that not all the pandemics are global pandemics. We have a lot of local epidemics, and having flexible local manufacturing capacity can really play a role there. Because pandemic preparedness is not just about developing a vaccine rapidly, but it's also making the vaccine available where it's needed. And this is one of the topics we're working on a lot with other stakeholders, obviously. We're working with other manufacturers, we're working with CEPI, we're working with the regulators to define a pandemic preparedness framework that is trying to take the most from the COVID-19 experience. And I'm going to tell you a bit more details in the next few slides. Now, regarding pandemic preparedness, I would like to take 2 steps back again, and we go back to 2020, when, even before we started clinical trials for COVID-19, there was this statement from the ICMRA, which is the coalition of regulatory agencies, and they were aligning among themselves on how to support the development of new COVID-19 vaccines. And they issued this statement where they basically say, if there's a well-characterized technology and you already have experience with that technology, you can use that experience to accelerate the development of your initial clinical trials for COVID-19 vaccines. This is suggesting the use of a platform approach. This is how we call it. And it means that you can really use the previous experience with different candidates and different products to help you understand what is going to happen with your new vaccine. And obviously, BioNTech at that time, we did have a lot of experience with messenger RNA in different formats and different lipid formulations that are key. So we were already working on messenger RNA candidates since many years, and we did already have a good wealth of clinical experience in different platforms. This is all oncology platforms where we were using the different formats of mRNA that we've also used for the early clinical development of COVID-19 vaccines. And you see the different types of delivery formulation from the RNA lipoplexes and the lipid nanoparticles, that is the formulations that we have tested for COVID-19 as well. So we had experience. How did this experience practically help, other than for our internal knowledge, obviously? We actually included this experience in the early regulatory documents for getting the first clinical trials for COVID-19 approved. So we leveraged that experience because it gave us, first of all, reassurance on what we could expect in the first clinical trials, but it also gave this reassurance to the regulators. And this has been key. So we used a sort of platform approach to extrapolate the experience from other therapeutic areas, other candidates into COVID-19. Now the question is, can we do that in other infectious diseases settings, taking the COVID-19 experience as a benchmark? To some extent, the regulators have started doing that. And this is what I showed you at the beginning, 2 months for the approval of BA.4/5 Omicron bivalent adapted vaccine. How was that possible? Actually, we didn't have clinical data for the bivalent Omicron BA.4/5 vaccine at the time it was approved. What we submitted to the regulators is a package including the clinical data for the bivalent Omicron BA.1 vaccine that was already available, manufactured before, and the pool of non-clinical data showing the immunogenicity of the BA.4/5 vaccine, and an overview of the non-clinical data showing the correlation across different variant-adapted vaccines between the animal models and the human model, and a safety data set composed of safety clinical data from different variant-adapted vaccines that we had brought to the clinic, alpha, delta, beta, and for which of them we could show consistent safety and reactogenicity. So at the end of the day, we basically established a platform where we could say, we know how adapted vaccines are going to behave. And this was the first time, actually, that an adapted vaccines was approved based on a package of data, platform package of data, and then clinical data came afterwards and confirmed the same. And this is the same process that we are following. This is an FDA presentation from 2023. We're doing the same in 2024. This has become the standard process for COVID-19 adapted vaccines, which is a sort of platform approach, if you want. We're trying to make this more formal. So we're working with CEPI and with other manufacturers and stakeholders to propose a pandemic framework that officially leverages the use of platforms to make sure that we can work with the regulators in a proactive way, establishing a pandemic platform beforehand that can be rapidly adapted to an emerging pathogen. Plus, we're trying to suggest some areas of improvement in the space of regulatory cooperation and regulatory harmonization. This is a consortium we have created with CEPI. You see other stakeholders that are involved. We created this last summer, and we are working exactly in these areas that I've mentioned. So, response playbook for pandemic pathogens, building on platform technologies from a regulatory framework perspective, and improving convergence and collaboration on key requirements globally. Now, my conclusions, very briefly. Obviously, the COVID-19 experience should be used and can be used to improve our ability to address pandemics, that's clear. It's a recent experience, we should really use it before we start forgetting about that. And the second topic is, it can be used in other areas in infectious diseases in different ways. Obviously, it was a proof of concept for messenger RNA, and it's important that we and others explore messenger RNA as much as possible, leveraging that experience, because it can be a game changer in a number of infectious diseases settings, but this is work that needs to be done. And we need new regulatory guidances and new definitions and a new regulatory framework around pandemic preparedness and around messenger RNA. My last message is that I've worked for quite some time in vaccines and antimicrobials, antifungals, antibiotic stewardship. There's one thing I've learned in these years that all these infectious diseases areas have in common, is that you cannot win alone. There's no way, whatever you do, whatever good your research is, whatever good your product is, there's no way you can be successful in isolation. We really need to collaborate. These are all areas that require collaboration, and sepsis is one of them. You can have the best product, you can have the best anti-infective. It's not going to do any good if it doesn't get to the right person in the right place at the right time. And the right place can be anywhere globally. So we need to be ready for collaboration. We need to be ready to share our experiences and to work more. I think we are making progress in the post-COVID-19 times. I see an improvement in global collaboration with the regulators, with other stakeholders, but we are not fully there yet. So this is work in progress. So this is all from my side for today. Hopefully we're going to have some time for discussion afterwards. I think we're a bit behind. I'm trying to be quick, but if we don't, you can reach out to me. This is my contact. You can ask questions. Thank you so much.
So this was wonderful, and definitely we will have time to discuss this, because I don't want to reiterate myself, and just ask Ole Wichmann from the Robert Koch Institute, who will add to this terrible discussion which we needed and were stressed by what was called hysteria. And just one note, when we handed over Professor Lauterbach as our Minister of Health the Global Sepsis Award, because he brought sepsis in the agenda of the G7. There were quotes in the internet that Himmler hands over to Goebbels the Global Sepsis Awards. So this is the scenery which is not backed on facts, but fake news, and hopefully you can clarify a little bit the balance between the risk and benefit of vaccine. Thank you so much.
Well, thank you very much for the introduction, and thank you to the organizers for the kind invitation. I mean, it's a big topic, and actually I would like to rephrase what the colleague in front of me who talked before said. I mean, we can have the best vaccines, but they are useless if there's no demand in the population, if people not get vaccinated. And so that's a little bit the topic I'm talking about here today. I think to talk about the benefits of vaccines, I think everybody here in the room and in the online probably are very much aware. So I will not spend too many slides actually on the benefits, because I think they're rather clear. I mean, this is just one example from the US with data with some diseases where you see the number of cases before vaccine introduction on the left-hand side, the achieved coverage, and then the cases in 2017, and you see for most of these diseases a decline between 100% smallpox is eradicated, and you see decline in diphtheria, for example, 100%, measles 99%, and so on. So tremendous success of the vaccines which are being introduced over the past decades. This paper has actually been shown already by Professor Kaufmann, but I wanted to stress here, I mean, this is we have birthday actually to celebrate this year of the expanded program on immunization. And you see here from a modeling work how many cases or death were prevented over the past 50 years. Here you need to bear in mind that initially the immunization programs, the expanded program on immunization, included around 6 or 7 antigens. So they're all directed infants. So what you see here is simply the number of infant immunization over these 50 years. However, especially after in the past 1 or 2 decades, there was an expansion of the programs. We nowadays also target pregnant women. We target adolescents, for example, HPV vaccination, which prevents cancer, and many vaccines are directed now against infections in adult, for example, pneumococcal disease, influenza, and so on. So huge development of new vaccines, introduction in the programs, and these new vaccines are not even considered in this analysis. So this is a slide that's more hypothetical, but to explain a little bit the problems what we are facing in vaccination. You see here on the left side, the pre-vaccine area, we have got high incidences of a disease. So everybody knew people around in their families or neighborhood who had, for example, case of polio or complications of measles. Then vaccines are being introduced, you see an increase in the coverage, and at the same time the disease is going down, as I showed, by 99%. So around yourself there's rarely anybody with a disease left. At the same time, with a coverage of 95%, I mean, vaccines, in some rare cases, they have got side effects, but suddenly you rather know a person who have maybe side effects, but not any longer the disease. So some people then say, okay, why should I vaccinate against a disease that's not anymore prevalent and causing side effects? But if you stop vaccinating, you see you have got the coverage is decreasing, and you will have outbreaks, and the disease will return. So that's a problem. What we call that the vaccines are the victim of their own success. And this is what we are facing here. I mean, this concept is good for some diseases, but unfortunately not for all vaccines. We can control a disease or eliminate. But here I give another example, and I used here the example of vaccination against influenza in pregnancy. I think I want to simply illustrate here some of the problems of misconceptions, not correct knowledge, and also the perceived risk. I mean, this study is 10 years old, but still, when I saw these results, I got really blown away and said, okay, this now really explains why we in Germany have a coverage against influenza in pregnant women of around 15%, 15%. So we recruited here actually pregnant women, 830, as a cohort study, and wanted to see over 2 years what is influencing their own decision-making on vaccination during pregnancy or afterwards for their child. And we ask here about knowledge, and for example, the first, that there are additives and vaccines that are not dangerous in humans. That's a true statement. But you see that only 43% said, yeah, I agree with this. 23% said it's wrong, and a large number, and that's also concerning, they simply didn't know. And the second, for example, here is that we said, okay, do you agree disease like autism, MS, or diabetes might be triggered by vaccination? And only 60% said, yes, this is wrong, that's false, and 43% didn't know for sure. You see that we have a large proportion who are not really sure what is true and what is not true. And then what I found really striking, then we also ask about the perception of in respect to the disease and to the vaccine. And when I say the risk perception, I mean what is the chance that you get the vaccine, the disease, and if you have the disease, that it's severe. This is one ranking, and we ask then the same for a side effect, for the risk of the vaccine. How likely is it that you have a side effect of the vaccine and that the side effect is severe? And you see for the majority of diseases, for example, measles, it was much higher perceived risk of the disease in contrast to the vaccine. But if you look below, this is influenza vaccination in pregnancy, suddenly we see that it's a much higher perceived risk of having side effects of the vaccine than a perceived risk of having influenza during pregnancy. I mean, this tells you all, I think, if there's a higher perceived risk of the vaccine in contrast to the disease, then you're lost and you will not have a high coverage. So there are actually many myths and wrong facts around vaccination. I could go here for hours into these statements. Here are some I highlighted here. Autism. I mean, everybody knew about this story about autism and measles vaccination. Vaccines cause infertility, can cause cancer. mRNA vaccines, for example, vaccination causes allergies. So many wrong statements around the internet, or that vaccines don't provide 100% protection. So it's really, there are many myths around. So I don't want to go really into every single issue or statement here. I want to focus on the one in the center. So the side effects and risks of vaccines are incalculable. People say we don't know simply, or that the vaccine is more dangerous than the infection. So these are common statements, and important for me to understand what is the process of vaccine development and approval. Many people believe, who are not really in the field, that there's suddenly a vaccine and it's being on the market, and that's it. Nobody is looking at this anymore. There may be side effects, but nobody really is looking at this. However, for each vaccines, there's a process in place which starts with discovery and preclinical studies, clinical evaluation in the different phases where you look at safety and if this vaccine works. We have got phase 1, 2, 3, and then there are regulators, either central or at country level, who look at all this data and approve a vaccine or don't approve a vaccine. And then once the vaccine is on the market, it's not stopping the evaluation or monitoring of these vaccines. Usually they're integrated in a national program. What we do in national programs, we look at the vaccine coverage, so exposure of the vaccine. We look at vaccine effectiveness and adverse reactions. So that's the common process for all vaccines which are then on the market. Here is one graph I have from the European Medicines Agency. So the different phases of vaccine development, but also afterwards, where vaccine safety is studied. So it's the whole process in parallel. Many people are looking at the safety, but there's also after vaccine is in broad use, because some very, very rare side effects you will not be able to identify in a vaccine trial with, let's say, 20,000 people. If there's adverse events of 1 per 100,000, you will not detect it. But you need to have systems in place once you have it in the total population, that you will pick up very rare side effects. So there's post-marketing safety monitoring, safety studies targeted, and so on. And this all is actually then feeding back into the evaluation decision processes. So if there's a rare side effects being picked up, there will be the regulators who are discussing this, if this is of concern, if there is still the benefit of a benefit-risk ratio which outweighs the benefits of the vaccine. And it's also the same for the committees which are actually recommending vaccines. So that is also another important step. I showed you before and after approval for vaccines. It's not only the regulators, but as a next step, there's also committees who are recommending a vaccine. So for the licensure, the regulators, they ask the question if a vaccine is safe and effective and of high manufacturing quality. But there are committees in each country and also at global level at WHO who are deciding how to make best use of the available vaccine in a given population. So beyond actually vaccine safety efficacy, they look at the local epidemiology, disease severity, population level effects, cost effectiveness, how is the best schedule, what are the target groups, and the implementation issue in the programs. There's an advisory framework. So at the global level at WHO in Geneva, there's the so-called Strategic Advisory Group of Experts on Immunization. So they are providing guidance on all vaccines. So there are also other technical advisory committees, like advisory committees on vaccine safety and vaccine standards. And so at global level, they recommend or they provide guidance whether to introduce a vaccine and to which populations this might be suitable and so on. But of course, it's at global level. So at country level, you need to consider if this is also the case for your own country. You look at local epidemiology. So at every country, this is also guidance from WHO, there should be national technical advice groups on immunization, so-called NITECs, who are then developing national policy and strategies, and who are also at least being involved in implementing a national program and monitoring the impact. This is a process, and I want to simply highlight, it's not only that there are sitting some people who are looking at 2 or 3 publications and that's it. So this is a framework that's being used by WHO SAGE at global level, or also a framework which we use here in Germany in our NITEC. It's a methodology of evidence-based medicine from the GREAT working group, where usually there's a systematic review which is being conducted, and then there are methodologies to see what is actually confidence that you have in this evidence on the data which are being identified. And then there are so-called evidence profiles on the efficacy and safety of a vaccines under question, and there are other factors like disease burden and so on, which are also then feeding in the final decision. and if there's a risk-benefit for this vaccine and whether to introduce it or recommend in a country or not. What is also important when it comes to benefits for vaccines, so it's not only the reduction of a number of cases of hospitalization, of death. That's the beauty of actually vaccines, that they confer much more beyond. And so these are the broader benefits. I mean, you heard about reducing disease complications, and which includes also, of course, sepsis. There can be preventing antibiotic resistance, which has been already mentioned. So these are factors which needs to be accounted when making this decision. But vaccines are also usually, you can consider the economics behind, because you prevent a lot of costs in the health system by not having these cases and the disease. So often these interventions are at least cost effective or sometimes even cost saving. You also minimize the impact on a family, especially in low middle income countries. You need to pay out of pocket for the treatment of the disease and make debts and so on. So there's big economic advantages, and also the social impact of equity, for example, or life expectancy and so on. So these are the broader benefits of vaccinations, which needs to be taken into account when making a decision on the benefits of a vaccine. So vaccines confer benefits to both individuals and the population. That's my main message here. So I want to not to go too much into the details, but just highlighting here that there are these evidence-based methodologies of these recommending bodies who actually look at these all this data. But important is not only to summarize and synthesize them, but also publish them, make this evidence available, but also provide a structure that shows the logical progression from the evidence to the decision. So there are so-called evidence to recommendation tools. This is, for example, used also by WHO. This is also used by other long-standing national technical advisory groups. Here are some of the criteria. I don't want to go into the details. I already touched on several of these, like resource use, equity, and so on. But criterion 2 is probably the most important on benefits and harms of the intervention. So this advisory groups specifically look at these data. And I give you just on one slide a recent example which we had in our National Technical Advisory Group, where we looked at RSV vaccines, which are now available for vaccination of adults. So what we did was a systematic review and also a modeling study. And in this systematic review, just as an example how it looks like, you highlight here what is the number of participants in the studies, how many studies were identified, what is the certainty of the evidence, which was in this case high, and then the relative effect of 0.25, which is an effectiveness of 75%. This doesn't tell you sometimes not too much. I mean, you know it's a 75% effective vaccine, but then relevant is the absolute effects. So in this absolute effects, there's also the incidence or the likelihood that you get the disease. The problem with these studies, so usually you simply take the placebo group and then you know it's 2 per 1,000. However, these studies, they were conducted during the pandemic. So due to the pandemic measures, anti-pandemic measures, there was not much circulation. That's the reason why we did some sensitivity analysis and then assuming, let's say, 10-fold higher incidences. So what is the risk difference between those who are vaccinated and not? It's 15 fewer per 1,000. So this gives you some idea about the absolute effects of the vaccine. And then we used a modeling where we estimated based on effectiveness data, based on the epidemiology, on the transmission of the disease, and assumption of 40% coverage, how many cases we will be able to prevent with this vaccine. So we estimated that based on these figures here, we prevent 2,200 hospitalization and 162 deaths over 5 years. And at the same time, there were a slight increase, or in one of the studies there were, I think, 2 cases of Guillain-Barré syndrome which might be associated with the vaccine. And so also in the post-marketing phase in the US there have been a couple of cases. So it's not yet fully established that this is a side effect, but just as a sensitivity analysis we estimated, okay, how many cases, if this is a true side effect, we would expect if we vaccinate 1 million people, and then we estimate this might be then around 10 to 25. So the risk-benefit ratio, what we look here, is preventing 162 deaths versus maximum of 10 cases or 25 with the Guillain-Barré syndrome, where usually this is not a disease which leads to death but can be long-lasting. So what actually can you do with all these issues about misinformation? There's actually, if you're more interested in this, a handbook on debunking. And so what would you do with this misinformation? If you expect misinformation, you can do a pre-bunking or inoculation, you provide warning. So this is when it's not yet established. So if there is already misinformation out there in the world, you need to know, okay, if this is largely unknown, then you probably keep on monitoring without debunking. But if there's already a framing is fixed on a misinformation, then there are methodologies, which is also science-based, of debunking. And just briefly to explain this, we use sandwiches. So what do you mean with sandwiches here? This is actually based on evidence. So this we call, and developed also in our institute, so we are here the National Public Health Institute in Germany, we call this truth sandwiches. So the aim is actually of supporting vaccinating doctors and medical staff in their education work, in discussion on vaccination, effectively debunking myths that are spreading around. So if you have in front of you sitting somebody who is actually having exactly this misinformation, how do you address this? How do you convince this person to get vaccinated? And this is, well, state the facts, simple, concrete, plausible, then identify the misinformation as such, the myth, and then explain where it comes from and how it misleads, and provide a factual alternative. If possible, point out the logical or the fallacies underlying the misinformation, and then reinforce and repeat the fact. So this is actually how you can address this misinformation. What we in our institute has actually developed a one-pager for several of these misinformation, in total 11, and we continue to expand this as materials which is provided to doctors. We are talking about risk-benefit ratio, the safety, efficacy, and several of these myths. And to summarize my talk, I also apply this technology here, and I want to stress that, yeah, risk and side effects of vaccines are monitored very closely worldwide. That's a fact. Vaccines carry less risk than the infection and illness they protect against. That's a fact. Myths or misinformation is that side effects and risk of vaccine is incalculable, and that vaccines is more dangerous than infection. And again, the facts. So the risk and side effects of vaccines are determined in clinical trials prior to their approval and following their continued to be monitored worldwide. And vaccine is only approved and recommended if its benefits substantially outweigh its potential risk. So that's my key messages, take-home messages. Thank you very much.
Thank you so much. I think we heard 3 excellent talks. And I ask all our speakers to come here, and we have very important discussions to have, how to overcome these myths. And as long as you take your seat, we need another chair, I think, or no, okay. I think what we have been missing, we have not used the opportunity also of this pandemic to explain the severity of infections in terms also that they may be life-threatening. And when I remember back to the campaigns by the BZGR, Germany looks and searches the Impfpass, which I think was not adequate. That's why I deplore still, and we need to overcome that in most countries, the word sepsis in the context of COVID-19 and in the context of influenza was not used. And that's why, and the CDC has acted. I don't know whether you have seen their last post. They said everybody can contract an infection. And most infections may result in sepsis. So, and they gave a hashtag. And this is, I would propose, I don't know whether you have seen this, because we still have to overcome that sepsis is somewhat specific, it's another disease. And this knowledge is not overcome. And if we can agree on this, I think we can increase dramatically the vaccination rates. And that's why we think that this agenda that we have come out was important. So that's my comment on this, and perhaps to me it was so convincing what we have learned from this pandemic. And if everybody of you comments a little bit in addition to the summary, and yeah, how we can overcome, because we have left this to the wrong group of people, to say the least.
I agree. And actually, I think it's you said something which is absolutely true. I think it's a broader issue, and I was thinking about that when you were presenting the sandwich, right? I think it's because this is the sandwich strategy is a reactive strategy, where there is misinformation circulating, and you try to proactively address it and debunk the myths. And there is a fundamental problem, which is, I think, even earlier than that, which is, first of all, the scientific literacy, which I have the impression it's an issue we have today globally, and in our countries in particular. I'm talking about Europe, but also the US. And it's not easy to think how to improve that, but it's certainly a question that we should ask ourselves, because people believe some things that they read somewhere, also because they don't necessarily have the knowledge and the instruments to understand what is a reliable source versus what is a non-reliable source, what information is presented in a scientific way, and what information is just myths and misinformation spread by someone. And the other topic is there something that we can do better in the way we, meaning everyone involved in vaccines, drug development, antibiotics development, authorities, is there something that we can improve in the way we communicate? And I was having this discussion 2 years ago, I think, with a colleague from EMA, and we were commenting on maybe some of the language that is normally used should change and should help people understand a bit better what we are talking about. And this conversation was about the use of the definition of the protection from COVID-19 vaccines that was misunderstood as something that was supposed to be sterilizing immunity, preventing transmission and infection whatsoever. Well, actually, we weren't expecting that. So, but people misunderstood that, and that became an argument, right? Like, I'm still getting the disease, why should I get the vaccination? That was not the point. The point was protecting against severe disease, protecting against death, hospitalization. But that became a topic, and so probably there is something we can all do in also communicating more effectively in these areas.
Well, I would raise a question. You don't mind. I mean, the fake news stuff over the last half decade or decade has increased, probably exponentially. And if you want to find it, you find a lot of fake news on social media. Is there any attempt by RKI or by PI or whoever, or on a European level, to counteract that by providing truth rather than fake on such social media, because I, at least, and the US election, obviously, which I follow closely, is an enormous example on that. Is there a way to overcome that by providing more truth on the social media, which, in my impression, have more impact than any newspaper nowadays? It was more a question, but the response or the discussion response would be, we need to do something on that level as one of many, but I think that's an impactful level.
Thank you for this question. I mean, definitely I agree with you. I mean, it's a challenge, and with social media, it's even a challenge much higher than 10 years ago. But the question is actually, who is addressing this? So there's also many players actually who could address this, regulators, public health institutes, and so on. But it's also a matter of staff and resources we have. And I know, of course, the WHO is working on this, and several other countries. They have at the National Public Health Institute staff who is trained in this. And but just to tell you, for example, in my unit, who is the immunization unit at the Public Health Institute, I've got 4 or 5 staff who's doing everything around communication of vaccines. And so if you really want to go into this business of addressing fake news in the internet, I mean, you need to do this continuously. Yeah. It's not sufficient once in a while to say something. You need to continuously work on this, and that's quite demanding. And just to add something, I totally believe that we public health experts have not always been perfect in communication. Definitely that complications of the disease like sepsis can be much better used, or for example, influenza and prevention of heart attack and so on. So there's more and more such data available which we could potentially use or should use for communication. On the other side, I would say, communication is not all. What I said, I mean, we used to have vaccines which are only targeting small children. Childhood vaccines, we are very good. We have really challenges in adolescents and adults. I mean, and here we have not the platforms or to reach adults, and this is what WHO is calling the life course approach. And here we need to have systems in place which can much better address adults who are the main target groups also for several of these vaccines.
If I got it right, that the physicians are the key people or professionals who are able to convince their patients on vaccination. And as long also the physicians in many countries, including the policymakers at the same time, are not really aware on the severity of many infectious diseases. So this is a problem. So I don't know the German, English word, there's a word of Lufthoheit, to communicate something. But so we need, again, also here an approach including BZGA, including the chambers of physicians, because we had one discussion with the BZGA 5 years ago, and the leadership at this time of the BZGA said, we don't want to make people afraid about Blutvergiftung and sepsis. And it's only half a year that you find something on the website of the BZGA on blood poisoning and sepsis. So there's a lot to do, and we will achieve it only with a joint effort. And that's what was our goal from the very beginning, mostly fostered by those who have lost their loved ones, like the Staunton family, like many families here and back and so forth. So that's, I think, our approach, and we would like to continue along this with your support, because alone, we won't achieve it, so that's it. I think for the sake of time, if you do not mind, because we need also a short break, I would say we would limit it to 10 minutes as a kind of bio break, and then start 15 minutes later. So with the next talk would be let us say by 12:10 or yeah. Huh?
Let's try 12 and it's 12:05.
Okay, so thank you so much so far, and I am impressed. Yeah. Dann fängst du.
Welcome back. We're a little bit delayed, we all know that, and therefore I will be very short to give more time to our excellent speakers of the next round. And the next speaker is Leif Sander from the Charité, the Department for Infectious Diseases, and he will tell us about the lessons learned on immunotherapy of severe infections and sepsis caused by or related to the COVID-19 pandemic. Leif, the stage is yours, and thank you for coming.
Well, thank you to the organizers for putting together such an esteemed event and on such an important topic. So I got the I would say challenging task to talk about immunomodulatory, that's what I call them, immunomodulatory therapy for severe infections and sepsis, and what we've learned from COVID. I think we've learned a lot. As you will see, I will cover some of the mechanisms and some of the challenges, some of the successes, but focus on what still remains, and not going into detail too much about all the different treatments, but giving you an outlook to where I think we should be headed. So this is the situation for many acute viral infections, but it was basically the situation we faced during the COVID pandemic, when this new disease arrived on our wards and units. Basically, you have a virus that rapidly replicates even in the pre-symptomatic phase, and at the time of symptom onset, it reaches peak replication, and then the immune response starts to kick in, so you can detect seroconversion antibodies and other markers of immune activation. And for most of the patients, symptoms would then slowly recede, and the majority would recover by around day 10. But for a fraction of patients, and those were the ones that we obviously are most concerned about in this room, they would move on and develop severe illness or critical illness. And that was at a time when you already had antibodies present, so the immune system was starting to control the virus. Viral loads were going down. Nonetheless, these patients would deteriorate and end up on our ICUs requiring organ replacement therapy, and oftentimes would not recover since mortality, especially among those on organ replacement therapy, was very high, up to 50%. This is how this would look like, and I think the next speakers will also talk about how we best treat patients in these different stages. Basically, you can see a dense infiltrate in the lungs, making it difficult for the patients to breathe, and sometimes impossible, requiring mechanical ventilation or ECMO therapy. So in those patients that did not survive the disease, we asked our pathologists to look into the lung, and we noted a severely disrupted architecture, because this is what a normal lung would look like, so a very delicate architecture made up of fine capillaries and alveoli to allow for very effective gas exchange. And this architecture is completely disrupted, characterized by hemorrhage, alveolar hemorrhage, microthrombosis, as well as a very dense infiltration of immune cells, myeloid cells, such as macrophages and neutrophils. And this is also something that prompted us and many others to investigate the role of an disproportionate immune response and how to best modulate this for the treatment of this very severe stage. Obviously, the symptom acute respiratory distress syndrome is very heavily investigated. Nonetheless, we have very few therapies, and this is taken from a review that was written before the COVID-19 pandemic. And what we know happens is that there is an acute viral infection or another infection caused by bacteria, or some other form of injury that leads to the loss of alveolar epithelial cells, can lead to damage of the underlining capillaries, and this leads to extravasation of fluid into the alveolar space. This is the white haze that you can see on the X-rays or on the CAT scans. and microthrombosis and influx of neutrophils and other immune cells that will cause further inflammation. And this obviously will not allow for sufficient oxygenation of the blood. So a group here from Charité, and we were part of this consortium, looked a little bit closer, and I think one of the mechanisms that we were asking what is happening, how is the endothelium damaged, how does it lead to a disruption of this barrier between blood and airspace. And a group led by Philipp Georg and Birgit Sawicki showed that basically through the circulation of immune complexes, because I told you antibodies have already been formed, they bind to viral proteins, can lead to the activation of complement, and complement was really one of the factors that was picked up in many screens, proteomic screens and other investigations. And the next speaker will talk about how to maybe target the complement cascade for treatment. And this could then lead to the induction of a certain type of immune cell, a cytotoxic T cell that can basically kill endothelial cells. It would lead to microthrombosis and actually facilitate further leukocyte infiltration and inflammatory tissue damage. I'm going to talk about this further downstream effects of the cascade, whereas the next speakers I think will focus more on a complement. Another important issue is during the resolution stage, or the attempted resolution, we will see that you have a repair phase, so an absorption of the fluid, but also an expansion of extracellular matrix and fibroblasts. And this can lead to a very severe form of ARDS, which has a very strong fibroproliferative component. And obviously, even though you resorb the fluid, you might not be able to restore gas exchange due to deposition of fibers and expansion of fibroblasts, and this can lead to fibrosis, a very severe condition, and a very severe form of ARDS. And I'm already telling you that during COVID we saw a lot of these cases. So one of the things we noted early on is that a dysregulation within the myeloid cell compartment that is often observed in sepsis, so this is a clear analogy to sepsis, was observed during COVID and is a hallmark of severe COVID. It's a paper we published early in 2020, capitalizing on single-cell sequencing technologies. We showed that in patients that have this more moderate form of the disease, so they do feel ill, they might have shortness of breath, but they do eventually recover, we see a very strong activation of monocytes, and these are characterized by certain markers that are sort of good for priming T cells and for sort of priming a favorable immune outcome. Whereas in patients that went on to require mechanical ventilation or worse, we saw somewhat of a dysfunctional phenotype of these monocytes with certain markers that I don't want to go into detail, but they have low expression of MHC molecules. They're not very good at priming T cells, for instance, and they're very poor at performing other functions that you would normally see, phagocytosis, exudative burst. And you also saw the appearance of dysfunctional neutrophils, or we could also call them suppressive neutrophils, because they have many markers that we are known to suppress immune responses, some of which are found in sepsis, some of which are found in the vicinity of tumors. So this was very striking, and it was a hallmark basically of severe COVID, and it has a lot of parallels with sepsis. I told you about the fibroproliferative phenotype of ARDS, which is very severe, is very associated with very high mortality. We wanted to investigate more closely what happens to the immune cells in the lung that I showed you. The pathologist basically visualized on slides from patients that had disease. We went into patients that were still intubated and analyzed their monocytes and macrophage infiltrations in the lung using bile fluid and again single-cell sequencing technologies. Again, skipping the details here, what we noted was an accumulation of a certain phenotype of macrophages in the lung, and this is also what the pathologist told us. And when we looked into the detail of what are the characteristics, the molecular characteristics of these macrophages, we noted a very striking similarity to macrophages that had been found in a very, very different disease, which is idiopathic pulmonary fibrosis. This is a disease that has an unknown cause, is chronically progressive. It mostly affects people aged 60 years and older. Many of the risk factors for IPF, in fact, overlap with the risk factors for developing severe COVID. And when you bioinformatically superimpose a signature that you find from patients with IPF onto the macrophages that we find accumulating in acute COVID patients, there's a very strong similarity, and we used many other statistical methods to basically show there's an immune response that is triggered by the host in response to the infection that resembles parts of the response that we find in patients with this incurable, basically deadly disease called IPF. So, and if we look in more detail, work with the intensivists and radiologists and so on, we can see many examples of patients. This is a healthy lung shown in a CAT scan that, they come in with this patchy ARDS, and then this will progressively consolidate and oftentimes lead to severe fibrosis, as shown here. This patient did not survive the disease. And if you score the lung tissue in patients that deceased from severe COVID, you will see that basically all of the lungs that the pathologist received for this cohort show signs of severe fibrosis. So fibrosis and inflammation driven by myeloid cells is a hallmark of severe COVID-19. Now keep in mind, all of these patients came down with the disease in a state where they did not have pre-existing immunity. This is different from after the rollout of the vaccines. It's something that we're intensely investigating now, whether and how this response actually differs now that we have pre-existing immunity. This is summed up here. You have an acute damage both to the endothelium, you have microthrombosis, and the damage to the epithelium that is caused by the infection. These monocytes, you can already detect them in the blood as a biomarker basically for severe COVID. There are other markers in plasma that can be used to stratify patients by severity, and many of these molecules are complement factors, and Martin is going to talk about that in a sec. You have the infiltration of monocytes and macrophages that rapidly differentiate into a phenotype that resembles profibrotic macrophages in IPF. We know that the virus in itself can trigger this type of response in macrophages, and we're investigating mechanisms, but this can finally lead up to what the pathologists then see is diffuse alveolar damage, disruption of the architecture of the lung, infiltration of myeloid cells, and deposition of fibers resembling fibroproliferative ARDS. So the question was, how can we target this? So one approach that we tried was to target the infiltration of monocytes by inhibiting their emigration from the bone marrow and immigration into the lung. Now, unfortunately, this was an investigator-initiated trial that fell flat in the end of acquiring sufficient funding to fully enroll. So I cannot tell you whether this treatment basically drug that was once developed for HIV treatment would actually work, but I think targeting monocytes and macrophages has gathered a lot of attention also in fibrotic diseases, and is something that should be further investigated in sepsis as well. Now the big question obviously is steroids, and steroids is, the question of steroids in sepsis and pneumonia and ARDS is as old as these diseases probably, but we got some very promising data on steroids in COVID-19, and this obviously came from the recovery trial, and this data came out in the summer of 2020, and from that point on, when these data came out, the whole world basically switched to treating patients with dexamethasone, which was the steroid used in this trial. Now what you can see is quite striking, and these results are unusual for ARDS trials. You can see patients that were on mechanical ventilation really had a reduced, and we're looking at mortality, a reduced mortality of about 30%. This is unheard of. Nonetheless, we're still looking at a vast proportion of patients that somehow did not respond to this treatment and still died. However, if you have more milder forms of the disease, do not give steroids. They can actually make the whole thing worse, because this apparently this response is not generally modulated by dexamethasone or steroids, but only the responses in patients with severe disease. We tried to look into detail how does dexamethasone actually work, and why do some patients not respond. Again, we resorted to our sequencing platform, and what you can see is that the monocytes that I talked about so much before are the cells that show the biggest change. If you put patients on steroids, the transcriptional changes in monocytes, so which genes they make, is modulated the strongest. B cells are also modulated, but I'm going to cut this short. B cells do not seem to mediate this effect that we see in this case with steroids. And again, I know this is probably very detailed data, but what I wanted to show you is that we had identified a signature that basically marks patients with severe disease. And now if you put patients on dexamethasone, this signature is again reverted to the level of patients that do not have the severe form of the disease, but the moderate form of the. So a reversion of a pathological phenotype in macrophages is mediated by dexamethasone. And if you look a little bit closer, and this is why probably it wasn't so easy to investigate how do these drugs work in patients with rampant inflammation, we had to use single-cell resolution technologies to find a minor state, functional state of monocytes shown here in turquoise that actually shows genes that are responsive, known to be responsive to steroids. So this is just goes to show that this type of technologies are really useful to find mechanisms of actions and to see in vivo drug target engagement in a clinical trial. And now I think this is the interesting part of the story. You can link the responsiveness on a molecular level to the clinical outcome. And this is, I think, what tells us how these drugs work. So we have this phenotype of monocytes that shows a dexamethasone response signature. However, they are much higher in patients that go on to survive the disease. And the clinical outcome can be 6 months later. Okay, so this is just going to show you that a molecular response phenotype correlates with the outcome months later. And the same is for the reversion. And this is the phenotype that we find in severe patients, and this is reverted, but only in those patients that go on to survive the disease. And this is again summed up here, that if you stratify by clinical outcome, you can see the dexamethasone-treated patients, they do revert, but only the ones that survive. So the ones that do not survive fail to molecularly respond to the treatment. And they probably belong to the group of patients that still die despite the correct treatment according to the guidelines. So you can even use these types of signatures that we derive from single-cell sequencing, obviously a technique that you cannot apply in really high throughput, and then you can accurately stratify other cohorts and basically predict their outcome early on. It's not something we want to do on a routine basis, but I think we need to do these types of things in clinical trials to be able to stratify patients to the right treatment. What we do at the time is a one-size-fits-all approach. I just want to acknowledge the people that did this study, specifically Florian Kurt, Joachim Schulze, Anna Aschenbrenner, and the two first authors here. For this was, I think, a very nice study that basically unravels the mechanism of action of dexamethasone in acute severe COVID, because it reverts a pathological signature in monocytes, which correlates with outcome. But I think it also paves the way for more precision diagnostics and stratifying. We want to do this obviously in a different way. This is something that we could only do in about 60 patients at this high level of resolution. And we are developing machine learning algorithms to basically scale this up using fewer and fewer parameters to be able to predict, will this person go on to develop this fibrotic phenotype? Will this person probably respond to treatment? Quick change, what about steroids in general? So Martin could also talk at length. So there are many trials out that use this in severe pneumonia. So the most frequent cause of severe sepsis, right? And there was a recent trial that came out before, that basically in 2023, and I think this might be the wrong the wrong citation here, that showed there is a survival benefit for patients with severe pneumonia if they receive hydrocortisone. We can talk about there were other trials that showed something else, and it goes to show, depending on the population that you select for your trial, the moment you start the treatment, the dose of the treatment, maybe the type of steroids, all of these things seem to be impacting this. The male to female ratio, all of these things need to be taken into account. This is the phenotype we saw with hydrocortisone in severe CUP. This is the phenotype we see with dexamethasone. Nonetheless, there's a big proportion of patients that we still cannot help with giving steroids. We do have, and this is something that COVID contributed, we do have data on other immunomodulatory treatments, most prominently anti-IL-6 and anti-IL-6 receptor antibodies. And we know that certain markers that correlate with IL-6 can predict a poor outcome. And I'm not showing you the data, but these antibodies can have a benefit even when given in addition to steroids. So some patients, probably the ones that fail to respond to dexamethasone, can respond to another type of treatment. So I would argue we need to better know this person is not responding to dexamethasone, to steroids, we need to give additional immunomodulatory treatments, and we need to know which one. We have new data on anakinra that it can have a positive effect. Martin is going to tell you about anti-C5a treatment, which I think is very promising. There are more broad inhibitors of cytokine pathways, such as the JAK-STAT inhibitors. And I think there's also quite a bit of interesting data coming out on other immunomodulators. Time constraints sort of prevent me from talking about all of this, but I think what I want to tell you is that the one-size-fits-all approach that we have been taking, we want to do evidence-based medicine. This is why we follow very strict protocols when we treat patients with sepsis, and I think this is good because it shows quality assurance. But in the end, this leads to this algorithmic way of treating people can lead to a very, one-size-fits-all approach, and probably we need much more precise and more personalized approaches to sort of save the rest of those that end up on our ICUs. And I think treatment and approaches like I showed you to better stratify the patients and use that in clinical trials as diagnostics while we test the drugs in a randomized controlled fashion to sort of know who responds molecularly and who does not, and how can we then devise alternative strategies, I think will also be very helpful for pharmaceutical industry to sort of better stratify and better select for their patients. With that, I'd like to thank you for your attention. If you have further questions, there's lots more to talk about immunomodulation and sepsis. Feel free to get in touch, and thank you for your attention.
Thank you, Leif. This is really clinical research at the top, and I think we will discuss that all later. Although there are probably a number of discussion points coming up immediately. Thank you again. Is my microphone on? Yeah. Thank you again, and we will now have a speaker via the internet. So this is a virtual presentation by Professor Evangelos Giamarellos, and he will talk about personalized immunotherapy in sepsis, and he will talk about the results from the ImmunoSEP trial. Here he is, at least, yeah, here he is. And thank you so much for attending this meeting, despite your busy schedule. And we are grateful that you are here and give your talk on this topic. The video screen is yours.
A great opportunity to be with you. I would like to apologize for not being able to be with you in physical presence, which has to do not with my busy schedule, but for a very difficult health problem in the family. And I apologize again for not being with you, and I hope that similar problems will not exist in the near and long-term future, so I can again be back with physical presence. So thank you very much for the opportunity to speak about the ImmunoSEP trial. This is my conflict of interest disclosure for this presentation. And I would like, first of all, to explain what precision means. And allow me to bring you a bit back to the COVID times and actually to give you 3 different cases of patients, all of whom have the same characteristic. They are intubated, they are under mechanical ventilation. But if you look at, although they appear, in other terms, phenomenically, the phenotype is the same. All of them have ARDS. If you look at their lab characteristics, each of them is different than the other. And it's not only the specific biomarker which is taken into consideration, but the combination of the biomarkers. For instance, take into consideration that the first one has normal lymphocytes, but highly increased CRP and ferritin, whereas the second has a moderate increase of CRP and increase of ferritin, and the last one has decrease of lymphocytes with increase of CRP and decrease of ferritin. How can this panel be explained? And why not all these 3 patients, they don't have the same characteristics? Could it be the case that the mechanism of the disease in each one is different? All of you are aware of the trial which ended in the approval of anakinra for COVID-19 pneumonia. And the idea was that when a patient arrives at the emergencies, you use a biomarker. The name of the biomarker is SUPAR. And the biomarker tells you that a patient who's phenomenally well, he's not under respiratory distress, will soon develop respiratory distress, but a respiratory distress which is guided by IL. And the primary endpoint demonstrates that after 20 days of start of treatment, just because the biomarker is increased, the patients are randomized into treatment with standard of care plus placebo or standard of care and anakinra decreases significantly the likelihood for worse outcome. And the importance is that when we speak about precision immunotherapy, what counts is the pathway. We should not limit ourselves into the traditional beliefs that I will not give the drug because this patient has a lot of comorbidities or low comorbidities. I will not give the drug because he's elderly or not. I will not give the drug because there is so much time which has passed since this time and the onset of sepsis. You see that in all the subgroups, the efficacy is the same. And someone may say, why is this so important? Because what counts is if treatment may change the trajectory of the patient. These are the trajectory of the patients treated with placebo and standard of care. In red are patients who, before start of the study drug, are hyperinflammatory. In blue, patients who had prevailing lymphocyte responses, and in velvet, patients with prevailing coagulopathic disorders. And you see that after 4 days, and after 7 days, all these constellation of genes which express and which culminate into the inflammopathic or adaptive or coagulopathic pathway are so fragile, they do not stay stable, and they change over time. But if a patient is correctly selected and treated in a precision approach, then the majority of the patients, they stay, in the case of anakinra, in a similar endotype over time, and the trajectory is limited. And in other terms, in our case, what is important is that the patients, they remain stable at the adaptive endotype. And in this logistic multivariate regression analysis, the only endotype which stays, the only treatment and the only variable which favors stability to the endotype is treatment with the precision approach. And of course, it is very important for the future that already two big regulatory agents, FDA and EMA, they have accepted this type of precision treatment for COVID-19, which means that the perception of the need for precision immunotherapy is already underway. In the past, what we were doing, all patients were receiving all treatment, no stratification by mechanism. And all of a sudden, in one of the old trials, which did not meet this primary endpoint and where all patients were receiving the same treatment, we discovered that there is a subset of patients who have both hepatobiliary dysfunction and disseminated intravascular coagulation. The investigators described post-hoc this combination as macrophage activation-like syndrome. And they post-hoc described also that this specific endotype experienced a huge survival benefit. So let's see what this macrophage activation-like syndrome is about. This means that the tissue macrophages are stimulated. They start reproducing IL-1 beta. IL-1 beta acts in an inflammatory approach at the level of the receptor of tissue macrophages, leading to further IL-1 beta production. And this IL-1 beta production leads to liver dysfunction, which is expressed by coagulopathy, but also extreme increase of ferritin. And this is completely different than the primary HLH syndrome. The other edge is what we call immunoparalysis. That means that from the onset of sepsis, if you isolate circulating blood cells and you stimulate it for cytokine production, you see that the first 24 hours, day one in our graph, they fail to produce cytokines. And all of a sudden, after 48 hours, that means day 3, in this specific situation, the monocytes of those who will survive, they wake up. They are able in simulation to produce TNF, whereas the monocytes of non-survivors, they're not able anymore. So the question is whether this situation, which may lead to failure to engulf bacteria and risk of secondary infections, and which we call immunoparalysis, is something that, if reversed, may lead to survival benefit. This led us to a series of studies. At first, a small phase 2A study with the acronym PROVIDE, and then a bigger EU-funded trial of phase 2 with the acronym IMMUNOSEP. What was the idea of PROVIDE? First of all, to see that indeed, in a prospective approach, these two endotypes, macrophage activation-like syndrome, immunoparalysis, exist in patients with lung infections or primary bacteremia, and how this classification reflects final outcome and treat the subsets of macrophage activation-like syndrome with anakinra. In order to do so, we implemented as tools ferritin, which at concentrations more than 4,420, that makes the diagnosis of macrophage activation-like syndrome, and ferritin below that cutoff, and in parallel, less than 5,000 receptors of HLA-DR on monocytes denotes sepsis-induced immunoparalysis. The intervention was IV anakinra treatment, 200 milligrams every 8 hours for 7 days. First of all, the classification. I need to specify that these are patients with septic shock. And this classification actually demonstrates that in what we call an average survival, these two extremes are associated with the worst outcome. If we go from down to top, from bottom to top, we have patients in red who are suffering from macrophage activation-like syndrome. They have the worst outcome, and they are roughly 20% of patients at septic shock. Among them are patients with a relatively better prognosis, but still high mortality, and they are roughly 42.9% of our classification of septic shockers, and they are suffering from immunoparalysis, whereas 37.1% are at what we call intermediate. When we focused our analysis in patients who had macrophage activation-like syndrome, treated 20 of them with placebo and 14 of them with anakinra, we found that by day 7, that means end of treatment, anakinra provides favorable responses. However, when treatment stopped, then this favorable response ceased to exist, which leads to the question. Let's repeat that at a larger scale, which means that patients with sepsis, if they have ferritin more than 4,420, which classifies them into macrophage activation-like syndrome, our assumption is that because this is driven by excess IL-1 production, we treat them with anakinra. If patients, they have ferritin below that cutoff, and HLA-DR is less than 5,000 receptors per monocyte, they are classified in immunoparalysis. And the assumption is that we need to stimulate immune response and to give them treatment with recombinant interferon. The majority of the patients in this scheme should stay unclassified. And this gave birth to a suggestion which we delivered at the EU, and we submitted that as a grant with the acronym IMMUNOSEP, a large phase 2B trial taking place in Germany, in Greece, in Italy, in the Netherlands, in Romania, in Switzerland, and enrolling patients with lung infection or primary bacteremia, all types of lung infection, community-acquired pneumonia, hospital-acquired pneumonia, ventilator-associated pneumonia. All these patients, they should meet the sepsis suite definition. They should suffer either from MALS or immunoparalysis, using as screening for MALS and immunoparalysis, ferritin and HLA-DR, exactly as I previously described. And what is mandatory is to start treatment the very first 72 hours from sepsis. The idea is not to randomize per drug, but to randomize per intervention and per strategy. So the one strategy is what we call precision immunotherapy. These patients, they receive standard of care treatment according to the Surviving Sepsis Campaign guidelines, but they get 2 more drugs, one IV and another subQ. In case they suffer from macrophage activation-like syndrome, they get for 15 days, 3 times daily, IV anakinra, and in parallel, every other day, subQ placebo. If they suffer from immunoparalysis, they get for 15 days, IV placebo and subQ recombinant interferon. And if the patients, they are randomized into the placebo arm, they get 2 placebos, 1 IV, 1 subQ, in other terms, 2 treatments, and of course, they receive standard of care treatment according to the Surviving Sepsis guidelines. And here we elaborate a different endpoint, which I see with a lot of enthusiasm, that all phase 2b studies, which are under development, are aligned, and they evaluate this endpoint as a primary endpoint, promising for efficacy. So the primary endpoint is what we call the change of the mean SOFA score from baseline by day 9. And according to our statistical analysis plan, this means that we calculate the mean SOFA score from day 2 to day 9, 8 values. We get the median of these 8 days, and we subtract for day 1. If a patient is discharged home before day 9, then we adjust by last observation carried forward. If a patient has died, then we consider for the days until day 9, which he's not in life, that the SOFA score is 24. And the secondary endpoints are how the primary endpoint looks separately in patients with macrophage activation-like syndrome and immunoparalysis. If the change of the mean SOFA score is prolonged by day 15, which is the end of treatment, mortality, of course, but also what is very important is to see if biologically this intervention is plausible. In other terms, does ferritin decrease in patients receiving IV anakinra? Is the count of HLA-DR receptors on monocytes increased under treatment with recombinant interferon gamma? And there is no doubt that we need to adjust. If indeed at the end of treatment, the infection, which is bringing the patient to the primary sepsis episode, is resolved or failed, or there is a superinfection. And superinfection has also to do with the safety. And there is no doubt that, as in every trial, we have predefined in our statistical analysis plan 3 main subgroup analysis of the change of mean SOFA score, of mortality by day 28, mortality by day 90 in subgroups of patients with patch 2, CCI, and SOFA score. So allow me to show you the progression of the trial. It's with a lot of enthusiasm, which I announce that this trial has ended. The last patient was enrolled in January 2024. The follow-up of the last patient was completed by the end of April 2024. We screened in total 672 patients, and we randomized 281, willing to demonstrate that in the very end, 1 of 3 patients with sepsis in our ICUs, they belong to either macrophage activation-like syndrome or immunoparalysis. Allow me to show you the flow chart of this trial. Indeed, 672 patients assessed for eligibility. And you see that the majority of the patients, they remain unclassified into immune state after measurements of ferritin and HLA-DR. And in total, 281 are randomized. And they are randomized into 146 to the standard of care and placebo, and 135 to standard of care and precision immunotherapy. According to the applicable laws in each of the 6 countries which participated in the trial, in total 5 patients, 1 in the placebo group, 4 in the immunotherapy group, after becoming resuscitated fully and regaining consciousness and being alive and leaving the ICUs, they decided to request removal of consent and data not being in agreement with their legal representative, leaving a final intent to treat group of 145 and 131 patients. I would like in this presentation, first of all, because this is the largest ever prospective trial-worthy entities of macrophage activation-like syndrome and sepsis-induced immunoparalysis are studied prospectively, to present the first teaching having to do with what are the characteristics of this patient. And also what is very important, and you see that in blue is the placebo group, in gold are patients receiving precision immunotherapy. And as a matter of fact, and here we split patients into MALS and immunoparalysis, that as a matter of fact, the values of severity scores, of SOFA score, Cholson's comorbidity, the age of the patients, the gender of the patients is pretty much similar irrespective of the state of immune activation. And this fully corroborates with what I started my presentation this early afternoon, that based on the traditional approach which we are using so far, it is impossible to tell who patient suffers from which state of immune activation, and we are in need of a biomarker. After discussion this morning with the consortium, we had, as a matter of fact, the trial is analyzed, but we did not have the time yet to produce the data in a visual format so that I can share as a slide the results with each and everyone. However, all of our consortium is pretty happy to announce that the primary endpoint for which so far analysis has been completed is successful. This decrease of the mean SOFA score by day 9 is achieved in 20% of patients in the placebo group versus 33.8% of patients in the active group, and the odds ratio of benefit under precision immunotherapy is 2.03 after logistic regression analysis, and the p-value of significance is 0.011. This is the consortium. These are our heroes. We started this journey in January 2022. We are extremely enthusiastic that our primary endpoint is positive. Actually, the analysis in a raw format was finalized just a few days ago, last weekend, and that did not allow us to present the primary endpoint in a graphic format. And we are kindly asking from the Global Sepsis Alliance to organize soon a webinar where we can present these positive results with all of you. But we are very happy to announce the positive outcome, and we believe that this precision approach is the start of a new era for sepsis management. Thank you very much for your attention, and I'm looking forward to take any questions.
Thank you so much, and we will come back to you later during the discussion time and your next talk, actually. Thank you so much. That looks so promising, and we keep fingers crossed that the results will be so get some impact in the near future. Next speaker is Martin Witzenrath from the Charité, the pulmonary department, and addresses the complement system in COVID-19 related ARDS and sepsis and the lessons from the PANAMO trial. Thank you. And I have to say, complement in immunology was almost always underestimated, and it's good to see it back on the table. Thank you.
Thank you so much, Stefan, for the introduction. Thank you very much to the organizers for inviting me and for having me today. It's my pleasure to be here. These are my disclosures. I'm going to talk about data from collaborations with Aptarion and with Inflarix, just to mention that at the beginning. Now, back in the early 2020, we thought that most probably the complement system might be involved in severe COVID-19, because from the clinical pictures that our esteemed colleagues from China and then later on from Bergamo in Italy reported to us, we thought, well, this is sort of a more or less typical ARDS, where myeloid cells are recruited to the lungs and activated, most importantly, neutrophils and macrophages, but also lymphocytes, as we then learned later, and where endothelial cells are activated and lung injury then occurs. So we, together with Leif Sander, Florian Kurt, many more patients, many more people recruited patients from the early on. The first patient was already included in PA COVID, a trial that with high granularity made observations and got biosamples from these COVID-19 patients. And when the first 40 patients were treated and biosamples sampled, Markus Reiser from the biochemistry department made a proteomics analysis and found out that the difference between those with severe COVID-19 and those with moderate COVID-19 lies obviously in inflammation and coagulation, but most importantly also in complement activation. Now, Leif Sander already deep dived into the interplay of complement and T cells that was new at that time, and to summarize that, obviously the complement system in COVID-19 leads to thrombosis and microangiopathy, as well as lung edema due to lung barrier dysfunction. Now let's take a closer look to the complement pathways. There are the three typical complement pathways, the classical lectin and alternative, that drive C3 cleavage. And as we know that in the end, the complement pathway leads to inflammation, tissue damage, microangiopathy, and coagulation. It could be wise to address C3 and to block this with an antibody, for example. However, there is further pathways important as are important contributors to the activation of the complement pathway, as for example the extrinsic pathway that is driven by thrombin, trypsin, and elastase that enzymatically cleave C5. And there's also the observation that inflammation, tissue damage, and microangiopathy themselves can increase cleavage of C5, and therefore it might be useful not to address C3, but maybe more downstream, maybe C5. But if you address and block C5, you would also block C5b or reduce C5b, which is the membrane attack complex that is important in the case of secondary bacterial infections to fight these bacterial infections, particularly on the ICU. We could also block C5a receptor with aftoralimab, but then C5L2 is still open, and so I think it's smart to address C5a, for example, with an antibody that's called vilobelimab. Indeed, in experimental models, C5a can increase inflammation in the lungs, as you see on the left panels, and C5a is increased in COVID-19 patients with pneumonia and even more in COVID-19 patients with ARDS. We thought it might be good to target C5a based on the hypothesis that SARS-CoV-2 infection induces inflammation of the endothelium and activates the complement system, that C5a also activates neutrophils, and we now know also macrophages and lymphocytes, causing tissue damage, and C5a promotes coagulation through tissue factor release. And then I already told you that there is this vicious circle where thrombin, plasmin, and other enzymes can further induce direct C5a activation. Now, vilobelimab is an antibody, a recombinant chimeric monoclonal IgG4 antibody that specifically binds to an epitope that is only displayed after cleavage from C5, so that C5b, the membrane attack complex, is left intact, and the antibody binds with very high affinity and blocks C5a biologic effects up to 100%. In March 2020, Niels Riedemann and myself discussed to start a phase 2 trial with this vilobelimab antibody in COVID-19. At that time, unfortunately, the ethics committee in Berlin was absolutely against any clinical trial in intensive care unit patients with COVID-19. So we had to move out of Berlin, and we contacted our esteemed colleagues in Amsterdam. And Alex Vlaar and his team then started the phase 2 study part of this trial. And they really did a great job in recruiting the patients within a few weeks, starting from April 2020. These were a total of 30 patients, 15 in each group, 10 each intubated, and 5 on high-flow oxygen. They received the antibody on days 1, 2, 4, 8, 15, and 22. And we discussed every single patient every week, we then found that indeed there was some sort of a lower mortality. 4 patients in the placebo group died and 2 in the vilobelimab group. But these 2 were interesting because one died when he was turned around to prone positioning and accidentally extubated. And the other one was meeting the exclusion criteria, as she had very, very severe COPD. Now, we also learned that fewer patients experienced renal impairment, and there was a faster reversal of blood lymphocytopenia in the treated patients and an early increase of D-dimer levels. We also saw that vilobelimab reduced C5a plasma levels very efficiently and very quickly, and this made particularly Inflarix optimistic, and so that they decided to go into a phase 3 study part, in which, in a double-blind, placebo-controlled, one-on-one randomized manner, 368 patients were randomized. The study was powered 90% to show 28-day mortality difference. It was conducted internationally, and patients were recruited from October 2020 to October 21. And the primary endpoint was 28-day all-cause mortality. I will quickly also look for the secondary endpoints. Most patients were recruited in Brazil and the Netherlands, some more in other European countries, in South America, South Africa, and Russia, and they were very well balanced between the groups. Now, the primary endpoint was met. That was a 23.9% relative reduction of mortality at day 28, which is the first pharmacologic treatment reducing ARDS mortality. And it was also reduced on day 60 by 22.7%, or in other words, 1 additional life was saved for every 9 vilobelimab-treated patients. We also found, and this was very intriguing, that the more severely sick the patients were, the more they benefited from the treatment. So patients with a PF ratio below 100, so severe ARDS patients, they had the most benefit as compared to patients with a moderate ARDS, PF ratio 100 to 200. There was more benefit in patients with lower GFR and with higher severity scale scores. So vilobelimab was more efficient in patients with higher disease severity. When we looked at the patients with only PF ratio below 100, again, the mortality was clearly reduced, significantly reduced by 32.8% relative reduction. Post-hoc analysis very recently then showed that, interestingly, those patients who, on top of steroids, I mean, almost every patient in the trial received steroids, but on top of steroids, some patients received tocilizumab, the anti-IL-6 treatment, or JAK inhibitor baricitinib, and those patients who received tocilizumab or baricitinib had even a better reduction of mortality as compared to those with placebo. So the effect of vilobelimab was better in patients treated with toci or bari as compared to the data I showed earlier. And interestingly, there was no, at least in this study population, no effect of tocilizumab or baricitinib on top of standard care in the placebo group. So vilobelimab was more efficient in patients treated with standard of care plus tocilizumab or baricitinib. There were no safety issues, particularly no increase in infection rates. And this brings me to the conclusion of this part of my talk, that the primary outcome measure in PANAMO phase 3 was met on 28-day, as a 28-day all-cause mortality was reduced. There was a statistical benefit of vilobelimab over placebo in a subgroup of severe ARDS patients. Safety was okay, and then vilobelimab received an FDA emergency use authorization on April 4, 2023 for use in severe COVID-19. But I think there are some urgent open questions, and I would like to pick two. First of all, the question is, do the PANAMO results matter anymore for COVID-19 in nowadays? Because the virus has changed, the immune situation in the population has changed, and obviously we cannot compare the patients that we see today with the patients that we saw in 2020 and 2021. And the second question is, do the PANAMO results and data from other studies support further trials in non-COVID-19 ARDS? And I would, in the end, briefly touch on that. There are data out on anti-C5a antibody vilobelimab in sepsis in a phase 2a multicenter double-blind RCT. 72 patients were investigated. At that time, there was no difference in mortality or vasopressor-free days or renal replacement therapy-free days, but there were two dosing cohorts that had higher ICU and ventilator-free days as compared to the placebo group, which makes at least optimistic. And now I switch gears and I come from the antibody to a Spiegelmer, completely different technology. It's an elaptamere. I don't go into the technology details, but this Spiegelmer in the end works more or less like an antibody. And in a murine cecal ligation and puncture model of sepsis, this Spiegelmer had shown that it is able to increase survival of these mice. And we did few years of work, spent a few years of work with the C5a Spiegelmer in murine models, and we first showed that C5a in pneumonia, general pneumonia, bacterial or viral, is increased, and it is also increased in our mouse model of pneumococcal pneumonia in the plasma and in the bowel fluid. Holger Müller-Radetzky did this work, and he showed that when he blocked with the Spiegelmer, the C5a in a model of severe pneumococcal pneumonia, lung injury was reduced. And when he put the severely sick mouse with severe pneumonia on our mouse ICU to intubate the mouse and to mechanically ventilate the mouse, and started the treatment with the Spiegelmer at the time of intubation, again, lung injury was clearly reduced. But not only lung injury was reduced, but also extrapulmonary organ injury, like liver injury, gut or renal injury. And I only show the liver injury pictures where you may appreciate that liver cells died in severe pneumococcal pneumonia, and NOx-D19, the speedomer, abrogated this effect, and this had something to do with obviously complement activation, with coagulation, and with netosis. So we, on the basis of all these data, started a early phase 2b study called ADCAP together with Aptarion. We are currently recruiting in 30 centers across Western Europe. The idea is to recruit 100 patients for analyzing safety, and most importantly for then being able to subphenotype the patients according to their clinical phenotype, but also according to their response phenotype. I don't go into the details here. It's a 2-to-1 randomized trial, IV infusion over 15 minutes. Participants are followed for 60 days. Enrollment started. We have currently around about 45 patients enrolled, and we hope to be finishing the enrollment by early 2025. Maybe just regarding inclusion criteria, we are addressing patients with severe pneumonia with a PF ratio below 300, requiring respiratory support, no matter whether it's high-flow oxygen or invasive or non-invasive mechanical ventilation. They have to have a CRP level higher than 50 and may not be on respiratory support for more than 48 hours at inclusion. With this, I'm coming to my conclusions. There is definitely some uncertainty regarding the causal relationship between the complement system and ARDS severity, but I'm convinced that the complement system plays an important role in ARDS pathophysiology. Trials targeting C5a in severe pneumonia, ARDS, sepsis are needed, and the burning question is whether we should include every patient or only specific subphenotypes of patients. Thank you for your attention, and I would like to thank all collaborators from Charité, Aptarion, Inflarix, and from the trials. Thank you.
The topic later in the discussion round. Our next speaker is Dr. Joachim Struck from 14.4, and he will talk about biomarker-driven, organ-specific novel therapeutic approaches in sepsis and shock-related organ dysfunctions. So.
Yeah, thank you very much, Professor Kaufmann, Professor Reinhardt, dear colleagues. I will start off just with saying, I mean, this is like carrying coals to Newcastle. That shock is really a big problem which can hit actually almost everyone. And it's a life-threatening problem, difficult to assess and insufficiently treatable. And there are several causes of shock, sepsis being the most important one, but there are many others which are just listed here in a schematic way. And if we come to numbers, these are numbers from the United States. So each year, up to 1.4 million patients are diagnosed with any kind of shock. Among these, 760,000 patients diagnosed with septic shock, 220,000 patients diagnosed with cardiogenic shock each year. And the mortality rate from septic shock, depending on the source of information that you're asking here, is around 34%. Mortality rate from the cardiogenic shock, the acute myocardial infarction caused cardiogenic shock, even higher with more than 40%. And if you count up these numbers, in septic and cardiogenic shock, you end up with more than 350,000 deaths each year in the United States. So it is really a big problem, and as we have heard before this morning, of course it would be best to prevent all this right from the start. Vaccination could be one tool, education is another tool, there might be others, but this is not the situation as of today, so we have to deal with these situations and have to ask the question how we can treat them. And we know that there are several pathways that we can lead to mortality in this situation. We just heard a talk about the complement system. There is the disturbed endothelial function, which is treated by my former company, Adrenomed, with a respective antibody against adrenomedulin. We at 14.4 follow another pathway, which is important in some 30 or up to 60% of the patients with shock. And the molecule that my colleagues Andreas Bergmann and Karine Bourgeois several years ago identified to play an important role here is a protease called dipeptidyl peptidase 3 or DPP3. This protease is known since many, many years, last millennium, 1967. And by the way, the EC nomenclature 3.4.14.4 led to the name of 14.4 of those who are interested in this. It is a metalloaminopeptidase which hydrolyzes dipeptidyl residues from the N-terminus of peptides, of small peptides, which can be 4 to 10 residues long. The protease is some 80 kilodaltons big. It's a monomeric protein, and it's constitutively expressed all over the body, essentially. It's highly conserved among species. The known natural substrates are angiotensins and opioid peptides, but there might be more. And in the physiological, in the normal healthy situation, this enzyme is mainly localized within cells, and only very low concentrations can be found in the bloodstream. So Andreas and Karine set out to develop first a test to measure the concentration of DPP3 in the blood. And as you can see on the left-hand side, you can measure with this highly sensitive test concentrations in the healthy or general population. But then if you look into and this is the initial findings that were published a few years ago now, in patients suffering from cardiogenic shock, in this case a mixture of different etiologies, ACS and non-ACS, you can find that patients having levels of DPP3 exceeding those in the healthy situation, meaning those above 14 nanograms per ml in this case, they have a much higher mortality rate than those with levels below. And what you can also see if you look at the shape of the Kaplan-Meier curve is that the people with elevated levels, they die quite fast, so within the first week of shock. So these were the initial observations from a cohort study that pointed to a role of DPP3 in these disease. Now in the meantime, and this is just a sketch to impress you, many cohort studies have been conducted measuring DPP3, not only cardiogenic shock, but also in septic shock and other situations. And what has been found in any of these situations that DPP3, elevation of DPP3 is prognostic of adverse outcome, circular failure and circulatory failure and death. And most recently, a large study with more than 4,000 patients in Switzerland was published. And this was led by Thomas Lüscher, who is now president of the European Society of Cardiology, and very recently published. And this demonstrated really a role of DPP3 in cardiogenic shock. And I only want to show to you some key findings here. So elevation of DPP3 is an independent predictor of the development of shock. And on the very left-hand side, you see the separation of high versus low DPP3 at baseline and after adjustment for many variables, clinical as well as laboratory variables, including troponin and NT-proBNP. elevated DPP3 is still a significant predictor of the development of cardiogenic shock. And the right-hand side demonstrates to you that this is a kind of steady continuous relationship. The higher the DPP3 levels above the healthy normal range, the higher the risk of developing cardiogenic shock. Now it has been tested whether this DPP3 adds information to what is currently used as a risk prediction system. There is a clinical score called the ORBI score, and if you would add the DPP3 information to that, you would find that this significantly adds information to the prediction of this clinical score. So elevated DPP3 also independently predicts mortality, 30-day mortality, as well as 1-year mortality in this patient set with an remarkable hazard risk here. And again, this is independent from all the information, clinical and lab information that's currently available. So Professor Lüscher and others are very much impressed of that, and they see a future of this biomarker already now for the risk stratification of shock patients. But we don't want to stop here. There's ideas on how this all comes. As I said, in the normal situation, DPP3 is located within cells. Now there are situations where cells break. This might be for several reasons, and then what was inside the cell gets out into the bloodstream. And there's thousands of molecules, but among these it's DPP3, which is now circulating. That's why we speak of cDPP3. And now DPP3 sees substrates, which it usually does not see because it's within cells. And one of the substrates is angiotensin II. There might be more, and DPP3 would degrade those, and as these peptide hormones are vital, this in consequence leads to myocardial depression and organ failure and eventually death. So this is not only a sketch, but it's really supported by experimental evidence. So what Karine has done, this is on left-hand side, she injected into healthy mice DPP3 that was purified from human erythrocytes. And what was measured here was the shortening fraction as a measure of the cardiac function. And you can see in the control, there's a certain level of the shortening fraction. If you would then inject DPP3, you can see that clearly shortening fraction goes down, so demonstrating that the excess DPP3 now in the circulation is detrimental to the cardiac function. You can turn this around if now right-hand side, if you look at this in again, a mouse model where heart failure was induced by injecting isoproterenol. This alone would reduce the shortening fraction. And if you would then add an inhibitory anti-DPP3 antibody, you can see that you can reverse this effect. Now, these data altogether really give the experimental proof of the mechanism of action here. The excess circulating DPP3 really causes myocardial depression, and this can be restored by an anti-DPP3 antibody. Now, this clearly has prompted us to develop this antibody as a drug now. And the antibody is a monoclonal antibody that has been selected from a bunch of several candidates. It was humanized, and we call it prosizumab, and this sketch demonstrates here how we think it works. It's applied, of course, intravenously. So from a dying cell, DPP3 spills out into the blood circulation. Now, if prosizumab is then present, it would bind DPP3 and prevent it from degrading angiotensin II, and thereby restores the control over the RAS system and normalizes heart function and thereby improves survival. Now, there are several preclinical studies that has demonstrated the efficacy of the drug. In rodents, I just don't want to show this here, because of the limited time, but want to show you some data which have very recently been published for a porcine model of circulatory shock. Now in these pigs, shock was induced by injection of feces, and then a standard of care arm was used as a control. So it was a fully resuscitated system where antibiotics, fluid, vasopressors were given on the one hand, and on the other hand, the second arm, there prosicumab was infused on top of standard of care. Observation was then for 12 hours, and several functions of several organs were controlled here. So under prosicumab, you can see that the circulatory function was improved. So the requirement for norepinephrine to achieve target mean arterial pressure was strongly reduced. It also improved the respiratory function. You can see here the Horowitz index that, as you can see, prosicumab treated animals have normal respiratory function. It also reduced the IL-6 expression. So now here we are with inflammation, our topic of the day. So it influences also this in a very positive way. And we've seen that also troponin levels go down under prosicumab or are not elevated at all. So based on these very promising preclinical findings, Professor Stefan Kluge at the University Klinikum Hamburg-Eppendorf was so intrigued by this that he said, I want to use prosicumab for a first in-human study on named patient basis. And so he included 3 patients who had a strong circulatory shock that were DPP3 positive. So now this is clearly a prerequisite. The patient must be DPP3 positive, only then the treatment with an anti-DPP3 antibody makes sense. And all these patients were judged by the physicians of having a survival probability for next day of only 10% or even less. So they were almost gone. And so the summary of the results is that prosicumab in these, I mean, we know it's 3 patients. It's not statistic here, but still want to share the data with you. Prosicumab was safe and well tolerated. At least, I mean, no drug-related severe events could be, or adverse events could be recorded. Efficacy, I will show you data on the next slide, and also for the mode of action. And the data are the following. If you see the upper left graph, this shows the activity of the DPP3 in blood samples. So it starts before the treatment with prosicumab at very high levels of DPP3 activity, and then upon infusion of prosicumab, it was a single infusion over 2 hours, levels go down into the normal range. And if you read it from left to right, next is norepinephrine need. It was reduced over time in these patients. Inflammation went down, IL-6 levels declined, and then you can also see that the Horowitz index improved. If you go to the left bottom graph, then further to the right, also renal function improved, and also lactate levels declined. So we just completed a phase 1 clinical trial, so very classical one. So this was a randomized, double-blind, placebo-controlled phase 1 clinical trial to establish safety, tolerability, pharmacokinetics and dynamics of escalating doses of prosicumab in healthy subjects. So this was done with Peter Pickers in Nijmegen. And so the trial has just been completed. It is formally not closed, so we're just about to lock the database, but I can tell you already that at least we did not see any safety or tolerability issues, and this will be published very soon. Just to give you an outlook on the program that we see, first point, CMC, very boring to the academics, but very important for the pharma industry. You have to have the drug, right? And in a quality, and we've completed this. So the colleague from BioNTech knows what I'm talking about. Preclinical toxin safety has been done successfully. There has been this named patient-based treatment, which I showed to you. Phase 1 is completed, and we now want to go for safety and efficacy in the target population that we're aiming at, cardiogenic shock patients with elevated levels of DPP3. So to conclude, DPP3 has been identified as a mediator which can cause shock when circulating in excess. DPP3's mechanism of action has been identified, and this is the degradation of circulating vital peptides, including angiotensin II. Elevated plasma concentrations of DPP3 are prognostic for shock development, severity of shock, and outcome, especially useful for risk stratification in cardiogenic shock. Procizumab, a humanized inhibitory anti-DPP3 antibody, has been developed. In animal models of shock, procizumab reduces DPP3 activity in vivo, leading to improvement of impaired organ function. Procizumab is safe and well tolerated in preclinical studies. It gave promising results in named patient treatments. It is safe and well tolerated in humans, and further clinical program is planned using a precision medicine approach. And I would like to thank my colleagues at 14.4, colleagues in Belgium, in France, in Zurich, Hamburg-Eppendorf, and Nijmegen in the Netherlands, and thank you for your attention.
Thank you. So it's amazing what a single molecule can do actually with this kind of amplification afterwards. Our last speaker for today is already known to us because he was already on the platform virtually before. And the last talk by Dr. Evangelos Giamarellos from Athens will be on biomarker-guided antimicrobial therapy for sepsis prevention. And it's the ACCESS trial he will talk about. It's you, you're at stage. Does he have
Thank you very much for the elegant presentation, and I would like to ask you, Mr. Chairman, to confirm that you can see my screen, and also if I speak loud enough to allow clear transmission. So I would like to move to another field, which is how is it possible with a biomarker, and also not just limiting ourselves to the biomarkers, but including in the biomarker characteristics of the phenotype of the host, we may prevent worsening of a patient with sepsis. This is what we call the ACCESS trial, which was published this year at Lancet Respiratory Medicine. And allow me, after I present my conflict of interest disclosure, to explain to you why it was so important to run that trial. None of us would argue against the fact that the major cause of sepsis in this world is community-acquired pneumonia. And if you look at the guidelines for the management of community-acquired pneumonia in the last 20 years, one major question is whether treatment with macrolides should be initiated as adjunctive therapy to beta-lactams. And over the years, there were guidelines which were suggesting in favor, and some guidelines who were suggesting against. When there is such a debate, what lies in between is that there is no real high-powered randomized controlled trial to demonstrate where reality is. And several times the guidelines rely on meta-analysis. This is the most recent meta-analysis, including most of the concluded studies. And as you see, the result coming from that is that if you add a macrolide with beta-lactam and you treat empirically community-acquired pneumonia, then you decrease substantially the risk of death. However, we are living everywhere now at an era of considerable antimicrobial resistance. And the question comes, resistance to macrolides everywhere in the world is roughly 50 to 60%. How is it possible to improve the outcome of the host? So if you look at the discussion of most of the manuscripts or published meta-analysis, they will argue in favor of a modulation of the inflammatory reaction after addition of the macrolide to the treatment regime of the patient with community-acquired pneumonia, and of course of severe community-acquired. But for a patient with severe community-acquired pneumonia who is admitted at the ward, the most important fact is if we can somehow prevent deterioration, in other terms, prevent mechanical ventilation and ICU admission, and provide early benefit. Early benefit has several elements. The first important element is to demonstrate benefit in the first 72 hours. The second is to have an attenuation of the intensity of symptoms. The third, improvement of organ dysfunction. And the fourth, attenuation of inflammation. And the question is, how can you achieve that? Is this possible to be measurable in clinical reality? When it comes to symptoms, the two big regulatories, the European Medicines Agency and the Food and Drug Administration of the United States, they use what we call the respiratory severity symptom score. In other terms, a patient is scored daily about the intensity of four main symptoms: cough, chest pain, dyspnea, and sputum. And patients who miss any of them, they are graded, they are scored for each symptom by 0. If it's so intense, making life unsustainable, it's 3. If they are in between, 1 or 2. In other terms, if you add them all together, the major points a patient may get is 12, and the minimal points, of course, is 0. I don't believe that there is anyone who's not well acquainted with the SOFA score, the importance of the SOFA score after the introduction of the sepsis redefinition, which is part now of the endpoint of all major trials. So if adding a macrolide to standard of care may improve organ dysfunction, SOFA score needs to be decreased. And how can we measure attenuation of systemic inflammation? But we do that daily. These are the randomized controlled trials, which have elaborated that for patients with community-acquired pneumonia, we need to screen the patients before treatment starts for procalcitonin, repeat measurement as the time goes on, and decrease and stop treatment early if we see a decrease of at least 80% or procalcitonin is less than 0.25 in case of community-acquired pneumonia. So, as a matter of fact, what we need is to run a randomized controlled trial in patients who have a specific phenotype, and in this phenotype, we include biomarkers, and we wish to adapt an early benefit after integrating all these 3 elements. Let's see how this can be done. How we profile the enrolled patient population. Adults with at least 2 signs of the systemic inflammatory response syndrome: fever, tachycardia, tachypnea, leukocytosis, SOFA score 2 or more, in other terms, they meet the sepsis definitions, procalcitonin more than 0.25, at least 2 of the symptoms of the respiratory severity score: cough, purulent sputum, dyspnea, pleuritic chest pain, and conformity to the diagnosis of CAP. That means positive chest X-ray, but also no contact with healthcare services for more than 2 days, the last 90 days. And then we randomize the patients. And they get standard of care antibiotics plus placebo, or standard of care antibiotics plus clarithromycin, 500 milligrams 2 times daily for 7 days. And as you may see, there is a very intense follow-up. So the primary endpoint should be composite, and it should contain how the patient feels about his symptoms. That means an at least 50% decrease of the respiratory severity symptom score the first 72 hours. And on top of that, the patient should also experience an at least 30% decrease of SOFA score, or to meet the positive criteria for procalcitonin. So the question is, in that trial, how severe are these patients? Before jumping into this, just to let you know that the intention to treat population was 133 in the placebo arm, 134 in the active arm. But look how severe these patients are. They're elderly. They have increased comorbidities. Their PSI is 120, well above the 91 threshold, which defines severe pneumonia. So all of them, they have severe CAP. Their SOFA score is around 4. The procalcitonin is well above 1. Almost 30% has pleural effusion. All of them have hypoxemia. All of them high CRP. And you see that most of them are receiving standard of care treatment with third-generation cephalosporins, beta-lactamase inhibitors, or piperacillin. And then the question comes. They are severe, no doubt, severe cancer. They are in the ward, none of them in the ICU because need of a ventilation was an exclusion criteria, but what percentage of them will get a parallel improvement in terms of symptoms, decrease of SOFA score, and attenuation of inflammation? It's 67.9% in the active arm versus 38.3% in the placebo group. In other terms, adding on top of beta-lactam clarithromycin provides an odds of early improvement of 3.4. Then someone may ask, could this be a bit subject to interpretation? Can we have something more hard to demonstrate the benefit? But if these patients, they had indeed severe pneumonia, a significant percentage of them should be deteriorated, should be in need of mechanical ventilation and ICU admission. Let's see the time curves of progression of the placebo group in blue and of the active arm in red into mechanical ventilation, start of vasopressors, and ICU admission. This is 17.3% in the placebo group, just 6% in the clarithromycin group. The hazard ratio is 0.32. That means relative prevention of ICU admission by 68%, and of course is largely statistically significant. And then someone may object and say, yes, early improvement, prevention of ICU admission. But what counts is to discharge the patient alive. And in order to discharge such elderly patients alive, you need to prevent a secondary infection. How often can all of us consider what we manage is to resuscitate the first sepsis episode, and then a patient may get a secondary infection with further increase of SOFA score, that means secondary sepsis, and then we lose the patient through that. Here are the time curves for secondary sepsis. 24.1% in the placebo arm, 33.4% in the clarithromycin arm. Again, in almost 50% prevention of the incidence of secondary sepsis. And when all these are met, then the likelihood for early discharge alive, the first 90 days, is increased by a relative 51%. But the question is why we call that an immunomodulatory treatment. It could easily not be a treatment having to do with the immune system. What type of evidence do we have? Allow me to bring you back to the COVID era. This is a manuscript which was published in the Blue Journal in November 2020, and there are comparisons of the intensity of the inflammatory reaction between patients with COVID pneumonia and patients with severe community-acquired pneumonia non-COVID. And as you see, in the COVID pneumonia, increases IL-1 beta, increases IL-6. This is not the case for IL-10. In the classical bacterial cup, IL-10 is up, IL-1 beta and IL-6 are down, which means that the classical bacterial cup is a pneumonia where the elements of immunoparalysis are prevailing. So if clarithromycin is an immunomodulatory treatment, by days 4, where benefit and the primary endpoint are met, this should be reversed. This is the expression and the production of TNF by mononuclear cells in the blood on day 4. You see that once you stimulate these cells, what you get is in the clarithromycin group much more TNF. And what is happening in the circulation? We realize that by day 4, with a more or IL-10 becoming undetectable. This is the first randomized controlled trial to prove the clinical and the inflammatory effect of one macrolide, namely clarithromycin in community-acquired pneumonia, a treatment associated with early reduction of the inflammatory burden, and in parallel to which radical
know the data also. He tried to get into the discussion also.
So if it's okay, we complete that. Actually, the data were actually overwhelming for the clarithromycin effects, and we will thank him later or now. Here he is. We had some issues, technical issues, so we come to an end with the presentation. We got the message, and it's extremely impressive, and start the discussion.
Stay with us. Please.
Stay with us, please. And excuse me? Okay, so we have to thank you so much for the very impressive 2 talks, and we'll go on to a general discussion, which I understand for technical reasons has to be internally. And thank you so much, and we give you an applause for these 2 talks. Okay. We are over time anyway, but we thought it would be worth to have a short discussion on these topics, which were, and I asked the speakers if they would be just kind enough come up to the board. I mean, the major issue for me is that I'm used to complexity, but it looks very, very complex, from neutrophils to monocytes, macrophages, complement, and then the small molecule DPP3 as kind of mediators to effectors. So we thought that we keep this kind of open for a short round of discussion, burning questions first, and then perhaps see what can we learn from all what we heard, mostly through COVID-19, not only obviously for the future of a better life without sepsis, which clearly is an ambitious goal. And if there are questions from the audience, I understand there is a microphone. Please just raise your hand and ask your question. Otherwise, probably the most experienced sepsis researcher.
I have just one question.
Will then start that.
To Dr. Witzenrath, because I wonder, there is this data, if you combine in this trial, in a sub-study, if you combine immunosuppressive substances, and perhaps you may elaborate on this because this is published on an abstract, and for me this was amazing, but I'm biased because I brought Dr. Riedemann from Hanover to Jena 15 years ago and he ran my ICU, so I'm biased.
Yeah, thank you for this question. I was intrigued when I was when I saw the data for the first time as well, and I showed them today, although I was afraid that you would ask this question, because actually I don't know. It's absolutely from my point of view, it's surprising to see that there is this extremely additive or even maybe synergistic effect. The only conclusion that I could draw from that is that there are two parallel pathways, and if you block both, you have an effect that you cannot see when you only block one, but we don't know yet whether this is true and whether this is the case.
Do you have a comment on that? Yeah.
I wanted to ask, what is the difference between giving C5a alone and C5a in the combination, if you compare these two groups?
What is the difference between anti-C5a and?
No, if you have one group with standard of care and C5a blockade, but no tocilizumab and so on, and then you have the group with C5a blockade and tocilizumab. I know you compared tocilizumab with and without the antibody, but what about giving the antibody with and without the suppression?
Well, I didn't show the data directly, but indirectly, as in the slide before that, you could see that the effect was maybe half the effect that we saw in this, but I have to say, very tiny subgroup in a post hoc analysis. Yeah, this might be the difference.
Which is around over 80% overall mortality reduction, and as
post hoc.
No question about this, but that is intriguing in some way, and there, as far as I understand, there's an interplay between C5, IL-6 and C5a, so they induce, IL-6 induces C5a, and if you block it, and it needs to be prospectively, of course, investigated. So this is my understanding.
So did I miss something? I haven't met micro. Yeah? Did I miss something? If you just stick for a moment on the C complement system, C3a would still be there. If you block C5a, obviously
C3a is still be there. Yes.
Is there a similar sequence initiated by C3a, or you just neglect that?
Well, C3a obviously plays a role in activating C5 to C5a and C5b, but it has also its own effects, and I don't think that we can neglect C3a, but obviously we don't need to block it as much as we need to block C5a.
Leif, you were if I understand fully correctly, you were talking a lot on the neutrophils' harmful effects. Is that a kind of accelerated granulopoiesis that then could lead to something I would call MDSC, neutrophilic MDSC, or I'm not sure whether this definition is very helpful, but still, is that a kind of a suppressive mechanism or kind of hyperactive mechanism mostly?
So one thing we didn't talk about, and it's important to mention that also in the context of vaccination, that obviously vaccinating against these viral diseases is very good in preventing the occurrence of bacterial superinfection, which oftentimes in flu and in RSV and in other infection ends up killing the patient. Actually also, this is what we will see in the future with COVID, is the same thing. That basically we will see COVID patients not dying from COVID. They get really sick from COVID, but they might die from bacterial superinfection. And one thing we found, but we have a very, I think, elegant mouse model that the induction of these suppressive-like cells, it's what basically, in this study we ran together with Martin's group, is basically predisposing the animals to bacterial superinfection. And now we have observational data from human that this might actually be the case. So it's not only that it's probably a physiological response to trying to sort of temper the inflammation, but it basically creates a breeding ground for bacteria. And you can antagonize it. And I know your group did work on that for TB. So this seems to be a common pattern, that you need to sort of, too much of a good thing can be really bad in these instances. So this is something, I think, for COVID, and this is where this all comes together with sepsis. All these viral infections will, in addition to the acute ARDS, be complicated by these secondary complications, such as bacterial superinfection or also cardiovascular events, where basically, these other treatments come in to prevent shock.
The most surprising, for me at least, is the DPP story, actually. This is always what I have dreamed of, a kind of reverse translation. First you have the active compound, and then you see it's a biomarker. Normally we have biomarkers and have difficulties to associate them with a effect, protective or pathogenic. You think that is but then you come with monoclonal antibodies, which I think again is it's a enormous step forward. So how would you recommend that or consider that in a broad application or approach?
Yeah, to a certain extent, I think we're actually proud of having resolved this puzzle, because everything goes together here. We have developed DPP3 as a biomarker. We have demonstrated it to be causal, of a detrimental situation, and now we have the tool with a monoclonal antibody to treat this. And I think what's really a key element, as I tried to mention, is really that we want to consequently apply the precision medicine approach. It means that it is a prerequisite to have a patient with an elevated DPP3 level. Only then an anti-DPP3 treatment makes sense. And what I think is new here, not only to this type of treatment, but also others, is that I think long-term we have to think not within the classical silos, I would say, of clinical diagnosis, but to think of certain pathways that are set on or not set on through different situations, to stay with the cellular injury. So, I mean, this can happen in trauma, this can happen in burns, this can happen in certain cardiological diseases and others. Not in all of these patients, but in certain parts. And that's why we need to measure first the meaningful biomarker, whatever that is. Ideally, it's directly mechanistically related to the drug. And this is what the situation here is with DPP3. Yeah. So we are focusing at first place, of course, to a first indication that we want to explore and that we are aiming to get a registration for, of course. But then we do see a huge potential to go into other indications as well.
Yeah.
Again, great data. I was wondering whether the test measuring DPP3 is that already something you're also developing, and is that already been is that already available? Like Stefan Kluge, could he measure this in his routine lab and then go ahead and put patients on treatment with compassionate use, or how does it work? Because basically, if you want this to be implemented, you need the test to be implemented at the same time. So you need to develop the two in parallel.
Great question, thank you. Actually, there are two types of tests that are developed at the regulatory level of CE mark. One is a classical kind of ELISA, which is not suitable for use directly on the ICU or so. But we have it also on the point of care platform. This is from companies Nexus in San Diego based, and this is on IB10 is called the analyzer, and there you have 20 minutes time to result, from the blood draw till the result. The machine is very small, like a classical, phone or something, relatively small, and this is what is currently available. But we are in the process of bringing this marker also on the big machines like the Roche and Beckman Coulter, Abbott, and so on that you have in the central labs. This takes a little bit of time because big companies are not the fastest. But this is, of course, our goal. We have it, but this is the key message. We have it available and will have it available when the drug hopefully gets marketing authorization. It will be possible to measure, and we expect it to become the marker also a companion diagnostics, because it will be, from a pure mechanistic point of view, it will be essential for the efficacy of the drug. Will be an interesting discussion with the regulatory authorities, by the way, on this topic.
Let's come to the elephant in the room, and that is obviously how can you reduce sepsis in the more general terms in the near future, and what did we learn from the kind of emerging disease platforms like the RNA. To me, sepsis sounds like the kind of the disease that's always there, and the reduction, I found out in my own experience, always takes longer if something that's been around for such a long time, even if the data are much more worse than for newly emerging threats. So if I may, you are the last. If I may go around, what would you think as the kind of the next steps, and if you want to comment also on it, not longer than a minute or two per head, so that we come around.
As I try to say,
in your head?
Yeah, I think, and Professor Reinhardt, I think, is always referring to this. What is very impressive is what the Australians have done, and this is really education. This is the most immediate thing that can be done to reduce really mortality, and this is the most immediate way. And from that point of view, I think this is the one that should be pushed the most. Vaccination is maybe a little bit more behind, but then again, I mean, as long as all this is not implemented, there will be still the need to have drugs to treat the disease when it's present, or the syndrome. But education is probably the first thing to do.
I absolutely agree. Education first, then vaccination, and then we need some treatments for patients. And I think addressing the immune system might be one way to go. So there's many things that we fail to do in the area of prevention.
in infectious disease for sure, but this goes the same for non-communicable disease. And let's face it, even though we have the best vaccines, we will not be able to prevent this disease, and we won't be able to educate everyone to prevent, the thing from progressing. And what my takeaway from all of this is, we need less one size fits all and more precise approaches and tools that are applied to the right patient, and then we'll get these impressive results, because this is probably a cascade that's activated here, and we need to go away from just, okay, this was caused by pathogen X, and this is why I give this, but you'd rather have to go from the molecular makeup of the individual patient and give the right treatment. And I think we're making progress in this area.
I understand you would think of a combination of biomarker or whatever endotype definition or characterization and more specific vaccine approaches in addition. Is that?
I mean, you can't vaccinate, people when they have abdominal surgery, they might have polymicrobial sepsis. There's no way we can vaccinate against everything, plus that we can have the best vaccines in the world, and a large part of the population is never going to get them. Either they don't have access or they don't want to take them. So we can't prevent this, and also in the ER, and things might develop very quickly, so you can't prevent sepsis, even though education is absolutely important, but we need to stratify the patients once they are on our units and then give the right treatments. And yes, this goes via biomarkers.
Thank you for this. I think we can only overcome the invisibility and the current under-resourced state of the global sepsis response to multi-stakeholder approach, and this has been said across the different presentations. If we can learn from success stories of immunization, HIV, malaria, or COVID, the multi-dimensional and multilateral approach made a difference. So that's why we were once again humbled with the possibility to launch this 2030 agenda that actually provides all the key approaches that we have to apply at the political level, ensuring historic investments in public, private, and academic partnerships, and the synergies of action between different stakeholders. We really hope to continue discussion on how we can make this a reality and really make a difference. Thank you.
I have a comment from the industry,
Thank you.
breakthrough industry.
Thank you so much. I think everything we said today, and we've seen some incredible data and very interesting insights as well, I think everything goes back to the fact that, as she said, right, we live in a world where nothing happens in isolation, and sepsis is a very good example of a complex issue that cannot be addressed in one direction only. So there are going to be probably very good treatments that are going to be, I don't want to say revolutionary, but potential breakthrough. And I think we need to be mindful that this is one piece that is going to help, and a lot more can be done in prevention. We were discussing in the panel before the importance of getting the vaccines that can be available, that will be available in the future to the right people when they need them. And this is an issue that we still have to solve. We were commenting about vaccine hesitancy and what does it mean. And so I think the bottom line message is really about collaboration across all the stakeholders involved, and all these activities need to be joint efforts, which means sharing experience, sharing data, and working across different levels of engagement as well.
So I would like to add what has been said, and what strikes me is, and again, it's an issue of awareness for a disease. And Axel Spriess has, after this 1.5-hour discussion in the Bundestag yesterday, at the very end, asked the question, how come that given this burden of disease, almost nobody, be it patients, be it professional societies, which are mostly, and this happened after World War II, because the question was, ironically, that the interest in infectious diseases declined which was determining whole medicine from 1900 to 1950, 60. And it was so successful as there's data, the crude mortality rate from infectious diseases in the US in 1900 was 800 per 100,000 population. It went down to the range of 50 per 100,000 population in the high-income countries. In Africa, it's still in some countries as high and higher as it was in 1900 in the US. So on what also, and this resulted in the perception that the fight against, and the book against infectious diseases can be closed. And I can tell you that in World War II, the British Army of Defense sent posters to every soldiers with big posters with the headline, "We're Hitler's best friend," called blood poisoning, killing more patients, has more patients as it's discredited as a joint allied forces together. Uses blitz methods. And do neglect yourself will help Hitler. So this awareness issue was there. The difference between the success of the COVID-19 also in Germany, where our mortality, our excess mortality rate is only half than this in the US and is similar to that in Sweden and is better or better than in UK. And the only difference I can see that whereas sepsis from non-COVID causes is twice as high in our country as in this country. And the only explanation is that when COVID was there, the whole society, including the policymakers, became involved. So we will not gain this fight if the high-level policymakers don't get involved. So, and now we have the tools, both in terms of developing and vaccination and prevention and education, of course, also the public, because there's data, and Dr. Sasse also has reported this for children, that 20% of cases, the parents don't recognize severity at home. And the same applies to adults because they don't know the symptoms. Everybody knows the symptoms roughly for stroke, for heart attack, etc. And that's why we need to ask and to invest in education, both on prevention, and we have half the vaccination rates against influenza and pneumococcal than in UK, in Australia, and the US. So this is one part of the picture. And as long as people don't know that sepsis is an emergency, and our campaign in the future will combine stroke, heart attack, and sepsis is an emergency. So that's one strategy. And that's why we call to professional societies, to policymakers on the country, on the Länder level and the national level. And only when we work jointly along such a strategy, and we have the proof now that things work. And the success against HIV is similar. The successes in cancer were similar. It takes time, but we lag back 30 to 40 years what other diseases, and they understood a disease can be transformed scientifically only when it can be transformed politically. And Rudolf Virchow has said, the physicians can only point to the problems, and it's up to the policymakers to address it. So, and this is still true today. And yeah, we have everything. We have the science, and we need to call sepsis when it's sepsis. And we need to agree that sepsis is a common final pathway of most infections. So everybody knows what cancer is nowadays. Everybody knows what HIV is. Yeah, but nobody knows that the own immune system, the exaggerating immune system, kills as a kind of collateral damage. And again, only if everybody has understood this, then we will be able to make a difference.
Thank you. That was impressive. But also, let's come back to the hope that it will change, and we will see that we learned a lot, and the last years clearly brought back infectious diseases on the agenda. And I'm not sure whether the fully understanding of the immune system can be expected, but you're right, it's a double-edged sword, and we have to look at all parts of the whole puzzle. But I would think that education, vaccination, whatever, prevention still is better than cure, and so let's try to get that on. And it's your part now to be influential in politics, and I'm sure you will do your part. No, let's...
No.
Well, others on that table also do a lot. Thank you. Thank you so much. Yeah. Thank you, all the people here.
Why don't we do families, one for midwives, and one for healthcare professionals in German. So thank you.